so any other people could see also the symposium. And again, a reminder that please ask questions through the preguntas y respuestas or Q&A button in the bottom of the Zoom screen. Today we have a very exciting program. We will go through different approaches of memory and diseases. And our first speaker is Professor Luis Ampuero. She's assistant professor of the Universidad de Santiago de Chile. She has a PhD in pharmacology, and he did a postdoc at the Universidad Nacional Andrés Bello. Her lines of research avoid effects of antidepressants in memory and interneurons and Alzheimer. So it's an honor for us as an organizing committee to have Estíbaliz here. She will talk about fluoxetine beyond the memories. Once again, thank you very much for accepting our invitation, Estíbaliz, and the screen is yours.
Thank you very much. Hello, everyone. I want to thanks Elias and Jose for the invitation. Appreciate the invitation to participate in this symposium. I heard a wonderful presentation in the last week, and for me, it's a great honor to show my results here. Compare the presentation?
Yes, go ahead and share your screen, please. It's looking good.
Thank you.
You should go to a presentation mode.
Here.
Perfect.
Well, the title is Fluoxetine Beyond the Memories. It's about the effect of fluoxetine or chronic fluoxetine administration in adult rats. The major depressive disorder is among the most prevalent disorder in the world. So as a lifetime prevalence in the world is rating in 8 to 12%, and also is more prevalent in women and people with low socioeconomic status. In Chile has a prevalence between 8 for men and 11 for women. And the World Health Organization estimates that in 2050, 46 million USA citizens will contract the disease, implicate a big cost for the health system for this country and of course for all of the world. The treatment of the depression including several drugs acting modulating the serotonin, noradrenaline, glutamate, dopamine, and melatonin receptors. For example, drugs like selective serotonin reuptake inhibitor or serotonin noradrenaline reuptake inhibitor or second generation of monoamine oxidase inhibitor or antagonist of glutamate receptor has ketamine or agonist of melatonin receptor has agomelatin are used for the treatment of depression. But we know that fluoxetine is the first line medication for treatment of depression, and it is one of the most prescribed antidepressant drugs. Fluoxetine is a selective serotonin reuptake inhibitor introduced in 1980 by Eli Lilly pharmaceutical company as used for treatment of depression, but also another neuropsychiatric disorder has bulimia nervosa, obsessive compulsive, post-traumatic stress disorder, and panic disorder, and showing high efficacy and tolerability in this treatment. The mechanism of action, classical mechanism of action of fluoxetine is inhibit serotonin reuptake, hereby increasing the level of serotonin in the synapses, but the antidepressant effect had a delay in the clinical response, suggested that this fluoxetine induced adaptive change in the circuitry and the synapses. For that reason, we assessed if a chronic fluoxetine administration induced effects on the forebrain, and we administrate fluoxetine 2 doses of fluoxetine 0.7 and 3.5 milligram per kilogram or saline, and we analyze the anxiety and learner's helplessness. We used a battery of behavioral stats to evaluate anxiety and learner's helplessness. As you can see here, we perform the novelty suppressed feeding and elevated plasmate to measure anxiety. As shown you in the graph, both doses of fluoxetine reduce the anxiety. This animal show an decrease in the latency to feed. As you can see here, the animal with high level of anxiety remain on the edge, but and the animal with low level of anxiety reduce or has a lower latency to feed. And in the elevated plasmate, we observe that only low doses of fluoxetine increase the time in the upper arm, suggesting reduce in anxiety. When we analyze the learner helplessness by 2 classical models to evaluate this behavior, we use a tire suspension test and forced swimming test. In both tests, we observe that both doses of fluoxetine reduce the immobility or reduce the learner's helplessness, but only low doses of fluoxetine increase the activity behavior as swim. For that reason, we selected only low doses of fluoxetine and we performed all experiment following. After the 4 weeks of treatment with fluoxetine, we isolate brain and perform coronal slide to immunostain it with several glutamatergic markers. As you can see here, representative image from limbic cortices as retrospinal cortex and prelimbic cortices show an increase in fluoxetine-treated rat of NR2A subunit or GLUAR2 subunit. This subunit is part of the NMDA receptor, and GLUAR2 is part of the AMPA receptor. We know that both subunits are important in synaptic plasticity. And the quantification showed that in the retrospinal cortex and in prelimbic cortex, we observed an increment in NR2A subunit, but also we observed an increment in GLUAR2 and GLUAR1. This result is in agreement with the homogeneous results we observe here that fluoxetine-treated rats show an increase in NR2A, an increase in GLUAR1 and GLUAR2, and we know if these changes are associated an increment in the postsynaptic compartment. We analyze postsynaptic density and we observe here that fluoxetine-treated rats show an increase in NR2A subunit, but now we observe a decrease in GLUAR1. Besides these synaptic changes were associated an increase in the colocalization of NR2A with PROSAT2 in synaptic membrane fixed on a slice we observe an increase in the colocalization with PROSAT2 here, an increase in the area of the punta. And moreover, when immunoprecipitated equal amount of NR1 and GLUAR2, we observe that postsynaptic density isolated for fluoxetine-treated rat show an increase in NR2A subunit, but now we observe and confirm that GLUAR2 is decreased. And these results show that AMPA receptor remain in an intracellular compartment, while NMDA receptor rich in NR2A is associated to an increment in the synapses. The morphological change associated to these synaptic composite changes were evaluated in this image. We performed a Golgi stain from pyramidal neuron in the limbic cortices. We observe here representative trace a different layer in the prelimbic cortices and retrospinal, and we observe that 4 weeks of fluoxetine or chronic administration of fluoxetine decreased the total dendritic length. And this result also was observed in the retrospinal cortex. Moreover, using another technique different to the Golgi stain, we perform electroporated neuron with MRP, and we analyze the dendritic length in the auditory primary cortex. Again, we observe that in fluoxetine decreased the dendritic length, the total dendritic length, and here we observe a decrease in the apical and the basal branches, which are the consequence over the structural plasticity induced by fluoxetine. We analyze the dendritic spines, and in the retrospinal cortex, we observe here that chronic administration of fluoxetine increased the synaptic density and increased the percentage of mature spines. Mature spines are characterized by bigger head, potentiated synapses, and rich in NMDA receptor. For other way, the fluoxetine reduced the percentage of immature that are characterized for thin spine and smaller, and not potentiation. Similar results were observed in the CA1 of the hippocampus. We observe here a pyramidal neuron in CA1, and we analyze in stratum radiatum the proximal dendrite to the soma. We analyze the dendritic spine, and we observe that fluoxetine increased the spine density, increased the percentage of mature spines, and decreased the percentage of immature spines. Importantly, we don't observe different when analyzing the dendritic spine in distal segment. Here is the point of, or the area they connect the entorhinal afferent, and this is important because the proximal dendrite receive afferent from the collateral Schaffer fiber. Which are the, in summary, we can postulate that fluoxetine induce a refinement of the dendritic arbor, induce synaptogenesis, increase the mature synapses rich in NR2A receptor, in NR2A subunit on NMDA receptor. The consequent functional on the of the chronic fluoxetine administration on the activity-dependent synapses, we analyze or we perform the electrophysiological recording, and we stimulate the Schaffer collaterals with high-frequency stimulation protocol, and we recording in CA1, and we can observe that only the control group was able to perform a long-term potentiation, while the group of rats with fluoxetine treatment, we don't perform the LTP, and in this case, the LTP was occluded. And if we perform a low frequency of stimulation, again, only the control group perform long-term depression, and the fluoxetine treated rat with, again, occluded long-term LTD. And in accordance with the morphological changes observed in the stratum lacunosum moleculari with no change in the spine density or dendritic spine density in this area, we analyze the response of stimulation, the temporal amniotic pathway. We stimulate this afferent from the entorhinal cortex and recording in CA1, and we now observe difference between saline or fluoxetine treated rats. In this case, the protocol performed LTP and LTD in the both groups. Which are the consequences with the cognitive function induced by chronic fluoxetine administration were evaluated, because it's important because fluoxetine is the antidepressant used for a long time after the remission of the depressive symptoms to avoid the relapse. For that reason, I consider important to analyze the cognitive effect induced by chronic fluoxetine administration. And we can evaluate the spatial memory for several behavioral tasks, like a Morris water maze and of the location memory. Here I show you the water Morris task. And this test, the mice is trained to reach the platform and using visual cues, and we train the animals for 4 days in 5 trials per day, and we measure the pathway and the latency to reach the platform. We perform the test 24 hours after, and then we record the latency and the pathway. Also, we perform the task 17 days after the training, and we observe the following results. We administrate fluoxetine for 7 weeks. We perform at 4 weeks, but the similar time to use it in a morphological experiment. We use it. We perform the Morris water maze and in 2 parts, 24 hours after training and 17 days after training. As you can see here, in the learning curve is similar to both groups. The latency to reach the platform is similar. And when we perform 24 hours after, both groups display normal result or similar results. But when we perform the spatial test at 17 days after, we observe that fluoxetine show increase in the latency, suggested that impair in the spatial memory, in the remote spatial memory. If these results are associated to a presence of fluoxetine in the blood, we perform the similar protocol, but now we administrate fluoxetine for 7 weeks and then perform the test 24 hours after training and 17 days after. In this time, we don't observe present or not represent presence of fluoxetine in the blood. As you can see in the graph, control group saline rats or fluoxetine treated rats show a normal curve of learning, show normal memory after that, but now 17 days after, the fluoxetine treated rats show an impairment in spatial memory. Now we retraining an animal to reach the platform, but in a new quadrant, and in this case, both groups display normal learning. These results suggest that the spatial memory is impaired in the fluoxetine treated rat, and we want to analyze the spatial memory for another task, less aversive, like object location memory task, and we administrate fluoxetine, and we perform this test in 24 hours after and 1 hour after the training. This test consists in train 2 mice to explore 2 identical objects. This phase is 10 minutes, so 10 minutes one day, and 24 hours after, we display one of the objects and maintain one object in the initial position. Here, we analyze the discrimination index. In this index represents the time to explore the moved object minus the fixed object on the total exploratory time. As you can see here, in the control group, display positive value of discrimination index, while the fluoxetine treated rat show negative value, indicate an impairment in the spatial memory. Then we perform 1 hour after training the test, and we observe that both groups respond normally to the discrimination index. That result confirmed the effect of a spatial memory induced by chronic fluoxetine administration. We know that the spatial memory is memory dependent on the hippocampal integrity. For that reason, and we observe that the fluoxetine modified the spine density and the dendritic arbor in the cortex, and we perform memory recognition task because this task is in addition dependent on the prefrontal cortex. we administrate fluoxetine again for 4 weeks and then perform novel object recognition task 24 hours or 1 hour after. This test is similar to the novel object, but now after training, we change one of the objects for a new one and maintain the other. In this task, the discrimination index represents the time to explore the novel object minus the familiar object on the total exploratory time. As you can see in the graph, the fluoxetine, so the control group rats, saline rats, show positive value of this discrimination index, while the fluoxetine treated rat show an impair in this recognition memory. And then when we perform 1 hour after training, both groups display normal value. These results indicate that fluoxetine also impaired recognition memory. We know that fluoxetine is one of the most prescribed antidepressants, and it is important to evaluate if these cognitive deficits are recovering after finally the fluoxetine administration. For that reason, we administrate fluoxetine for 4 weeks, finish the administration of drug, and then allow it a withdrawal period of 6 weeks. And 10 weeks, we perform the same paradigm, novel object location memory, novel object recognition memory, and Morris-Watermain task. And as you can see here, the spatial memory evaluated for Morris-Watermain or overlocation memory task perform normal value, indicate that this memory has recovered spontaneously, but now we observe that the novel object recognition memory is still impaired, suggesting that we need more time to observe the spontaneous recovery, or this is a deficit that is still permanent or persistent. How fluoxetine work to mediate the antidepressant effect is shown here. This is our results obtained for my laboratory, and also is obtained for several groups in the world, and is related to increment induced in BDNF signaling by fluoxetine. Fluoxetine increased BDNF and increased TrkB content in the homogenate with a delay compatible with the antidepressant effects. We know that BDNF is a neurotrophin secreted by synapses in an activity-dependent way. BDNF bind TrkB, and for this signaling mediate the several cellular function. BDNF participate or is involved in neural differentiation, survival, and synaptic plasticity. We know that the BDNF signaling increase after fluoxetine administration, but we know also that the TrkB receptor decrease in the synapses. These are the adaptive change induced in the BDNF signal. When immunoprecipitate, or when we perform Western blot of subcellular function, we observe an increase in BDNF and TrkB in synaptosomes, but decrease in the synaptic memory. Finally, an important work for one of the most important group in the world in the study of fluoxetine and BDNF, they describe that fluoxetine mediate the BDNF effect. Here they show that transfected neuron with GFP TrkB, the BDNF administration or fluoxetine increase the presence of TrkB in the membrane, and this presence is inhibited when the TrkB has a mutant in the tyrosine residue 433. This residue is important to BDNF signaling, stabilize TrkB in the membrane, and allow the BDNF signaling of TrkB. we observe here that this residue is important to fluoxetine bromodioxyuridine incorporation. We know that fluoxetine induces proliferation, and this proliferation is blocked when in the mice that has a TrkB mutation. Also, the antidepressant effect induced by fluoxetine was blocked in this mutant. As you can see here, the immobility is reduced by fluoxetine, and this effect is blocked by when in the mice that has this mutation. In this work, the author postulates that antidepressants like fluoxetine, imipramine, or ketamine increase the TrkB in the surface and the cell surface, stabilize the TrkB in the membrane, and allow it the BDNF signaling in an activity-dependent way. And in summary, we can postulate that fluoxetine induce a maturation competitive state characterized by a refinement of the dendritic arbor, increase in the synapses, but this structural change as a consequence in synapses with less plasticity in the synapses. We observe an occluded LTP or LTD and remote memory impairment. But for spatial memory, this impairment spontaneous recovery after 6 weeks of withdrawal, but the recognition memory not occur the same results. This is the same. Thank you very much to hear me. And this is my actual collaborator, la doctora Úrsula Wieneke in la Universidad de los Andes, Brigitte Van Sunder y Lorena Varela in the University of Andrés Bello. Thank you very much.
Thank you, Estivalis, for your great talk. Great talk. Nice work. As a reminder, we have questions from the audience. There's one from Paulina Hardy. The question is, what are the implications of evaluate fluoxetine effects in control animals? How can you translate your findings to a depressed animal, knowing that depression animal models have memory and morphological alterations?
The morphological alteration for stress or for depressed animals is in the same way. We observe that the stress and in a stress animal, we observe an atrophy of the cortical neurons and hippocampal neurons. And we suppose that in this case, the atrophy or refinement is complicated results for the antidepressant effects. But we know that this dose obtain antidepressant effect, and we suppose that the structural plasticity induced by fluoxetine recover the antidepressant effects, and show this similar morphological in the same sense or in the same way.
There's another question from the audience, but I guess it's in the same line than the previous. Would SSRIs that reverse depressive-like behaviors have the same synaptic changes and memory effect that you present in your study on animal models of depression? Have you learned or read about that? What happens in depressed animals?
In depressed animals is chronic stress animal. And in chronic stress animal, we observe the same effects by the stress. And for this reason, and we suppose that in this case, it's complicated and analyze the effect of the behavior, because the behavioral or the memory are associated for the effect of antidepressant and for the stress protocol. And always is important to evaluate in the stress animal, but this is not the interest for me because the stress induce a deficit in memory, and we observe maybe more higher results in spatial memory or other memories.
Estivalis, from what I learned here is that fluoxetine makes you happy because it makes you forget. Something like that. One thing I didn't get, excuse me if I'm not asking a real proper question here, but you show us that fluoxetine induced maturity of spines in layers of CA1 neurons, if I don't remember. But this does not lead to neither LTP nor LTD. Shouldn't we expect that mature spines promote LTP and LTD or not?
I suppose that the protocol of LTP and LTD as are occluded in these spines, or in these spines that are bigger and more NMDA receptor has less AMPA receptor. For that reason, not is able to, in this state, induce LTP or LTD. But the animal learning in the normal condition. And we suppose that other synapses can perform LTP or LTD.
Okay.
But it's specific learning.
Okay.
I have a question too.
Please go ahead, Francisco.
Yes, hi, Estivalis. Thank you for that excellent presentation. So I was thinking if, if like someone else has done, like what the effects of fluoxetine will be on like memory extinction. Because that could also be helpful, like while memory impairment is like bad in some senses, like forgetting bad memories could like actually be a helpful thing.
But in memory effect of fluoxetine, we observe effects dependent on the age of the animal, depend on the dose of fluoxetine used, and depend on time for the treatment. And one of the authors important in the field, they show that fluoxetine increase the extinction and of fluoxetine or extinction memory. And we suppose that high dose of fluoxetine maybe can increase or improve the memory, but the clinically relevant dose of fluoxetine used by my experiment low impairment in the remote memory. We observe, we don't realize experiment to evaluate the extinction memory, but we suppose that this result is observed.
Thank you.
Anyone else can ask a question? Remember to do it through the preguntas y respuestas or Q&A button in the Zoom screen.
I have a question. You find that the perforant path have no effect when you treat the animals with fluoxetine, but when you test the CA1, CA3, LTP, you have the difference. So what do you think that is the relevance of this difference between both effects?
The connection of the circuitry from the entorhinal cortex to temporal or the afferent pathway, the perforant pathway to a CA1 is only from layer 2 and layer 5 is to the dentate gyrus, and the layer 2 or 3 is to the stratum lacunosum moleculare. I suppose that this input from the hippocampus is maintained, and the Schaffer collaterals are in this state of mature compatibility state, and in this input, the long-term potentiator has a deficit. And the importance of maintain a gate, o sea, te lo voy a decir en español mejor porque me estoy confundiendo. Entonces, yo creo que la importancia de mantener una entrada desde la corteza entorrinal hacia el hipocampo en condiciones de salud sináptica, en el fondo, en la posibilidad de que tú puedas aplicar un protocolo de potenciación y observar la respuesta, permite que los animales y las personas que se administran con fluoxetina puedan aprender, porque finalmente una gran conclusión del trabajo es que el aprendizaje no se ve afectado, pero memorias evaluadas en un curso temporal muy específico están dañadas. Entonces, eso permite que en el fondo nosotros observemos que tanto las ratas, o sea, que las ratas tratadas con fluoxetinas pueden aprender, pero que si nos esforzamos más o hacemos un poquito más restrictivo el análisis, ya observamos déficit en memoria remota en estos animales. Entonces, es algo bueno, es algo bueno también que revierte espontáneamente, y me imagino yo que la memoria de reconocimiento también revierte, pero necesitas un tiempo más importante, ya que esta está almacenada en la corteza prefrontal, que es donde nosotros observamos cambios. Ahora, yo estaba hasta antes de la pandemia evaluando los cambios morfológicos, si es que esos eran reversibles. Yo espero que sí, pero no lo pudimos hacer. Así que espero que sí, pero no lo hicimos, y eso. No sé si tengo...
Gracias. Gracias. Entonces, tengo otra pregunta que es relacionada básicamente con el cambio que tienes entre los receptores AMPA que permean calcio a AMPA que no permean calcio. Tú mencionaste algo de BDNF podría estar involucrado en esto, pero ¿cómo sería el efecto de fluoxetina? ¿Lo haría directamente sobre la vía TRK o lo haría sobre algún otro receptor?
No, pues se supone que el efecto de fluoxetina es mediado por la señalización con BDNF. Entonces, fluoxetina se une al receptor de TRKB, lo estabiliza y permite la señalización dependiente de actividad de BDNF. Entonces tú necesitas BDNF o necesitas la actividad sináptica para inducir los cambios adaptativos mediados por la fluoxetina. Entonces, finalmente tú vas a observar que estas sinapsis que están más grandes, ¿cierto? Porque fueron estimuladas dependiendo de la actividad sináptica, pero estas sinapsis no pueden cambiarse. Entonces es menos plástico el sistema, porque la dejas en una condición que está como más rígida en el cuanto a nuevos cambios. No tiene tantos receptores, no tiene receptores AMPA para que respondan y produzcan grandes depolarizaciones, o tiene menos receptores AMPA para que produzca una nueva potenciación en esa sinapsis.
Gracias.
Any further questions? No, I think that's it for questions. Estivalis, I would like you once again, and thank you for one, accepting our invitation, and two, for giving us this amazing talk. It's been a pleasure to have you here.
Thank you very much.
I would ask you also that if you could turn on your share screen, please, so we can. Thank you. We will make a short break. We ended up this talk a little earlier than schedule. So for now, we're gonna make a small break. We'll be, as stated in the program, at 10 a.m. with Dr. Alexia Nunez Parra. So please stand by, get yourself a coffee or something, and we'll be back in a few minutes. Thank you. Hi, welcome back, everybody. Thank you for your waiting. We will continue to our meeting. We have now Professor Alexia Núñez-Parra. Alexia Núñez-Parra is a biochemistry from the Pontificia Universidad Católica Valparaíso. She has a PhD in neuroscience and cognitive neuroscience from the University of Maryland. And currently she is an assistant professor at the Department of Biology at the Faculty of Science at Universidad de Chile. Her works try to understand how neural circuits process sensory information and how neural coding is altered in neuropathologies. She's focusing now on how neuromodulators regulate the excitability of neurons in an in vivo murine model and how signals processing lead to hypersensitive in a model of autism and intellectual disability like the Fragile X syndrome. Alexia, it's an honor for us to having you here. Once again, thank you for accepting our invitations. One few reminders for the audience, please remember that you can make all your questions through the Preguntas y Respuestas or Q&A button in your Zoom screen. Do not make it through the chat because the chat is for other purposes. We are going live on YouTube, and all these exposures will be there for you to see them in offline or in another time. So don't be sad if you could not see them all. Once again, Alexia, thank you for being here. It's an honor, and the screen is all yours. You have to share screen though. Perfect, it's looking. Perfect. We don't hear you.
Yes, now I mute myself. Well, yeah, can you see the screen?
Perfectly.
Perfectly, okay. Well, thank you, Jose. Thank you, Elias, for inviting me to this symposium. For me, it's a pleasure to be actually here because when I first moved from the US to Chile, back to Chile, and I was starting my first or second year as a junior PI or principal investigator, I actually attended Neurosur at Universidad de Chile, and it was a really, really nice experience for me to be able to have like an opportunity to interact with all the researchers here in Chile and all the people that you had invited. So for me, it's a pleasure to be here now, giving this talk and having the opportunity to share this data with you guys. So thank you, and thank you to the organizers for all the efforts that I know they put in organizing this Neurosur Symposium. So, okay, so first when Elias asked me to give him the title, I just said basal forebrain and decision making, but now I add a little bit of information to the title, and I'm now put basal forebrain and auto guided decision making. It made more sense to be more specific about this, and now you're going to see why. So let's see here. So as Jose said, my laboratory seeks to understand how the brain interprets sensory information and how neuromodulators actually can shape how individuals perceive the environment and how they adapt to perform adequate decision-making processes that ultimately, of course, affect behavior. So in general, how perception works, and in a very simplistic way, is that we have an external cue. It can be a chemical cue, an electromagnetic wave, and or any different physical stimuli that actually get into our sensory organs. the sensory organs, such as the olfactory epithelium in the nose, the retina, photoreceptor, etc., transmute that physical information into electrical activity, and after that they transmit that information as an action potential to the central nervous system, to different regions that are going to process and code and decode that information step by step. In each of these lower processing regions, information is going to be modulated and shaped until it reaches the cortex, where information is mostly integrated, and integration between bottom-up and top-down information actually occurs. And then sensory perceptions begins, or it's executed. And after the world is perceived, or that particular sensory cue is perceived, then we can expect that the individual will have a behavioral output. But of course, this is not linear. We know that individuals behave depending on context, depending on the internal state, depending on several different information that the animal is actually integrating, and that occurs because we have a lot of feedback that actually talks between behavior, higher processing regions and lower processing regions, and that feedback actually is what drives my curiosity in the laboratory, and we try to understand how does top-down regulation actually shape how incoming information is perceived by the animal. And to do that, we use the olfactory system as our experimental model in rodents. And that's because the olfactory system has several features that make it really interesting to study electrophysiology and how we can actually combine electrophysiology with behavior. So here in this screen, you actually have, this is a rat head, and here you can see a mouse brain. This is the olfactory epithelium right here, that where the olfactory sensory neurons are located. And this is the, if you can see here, this big ball, this big protuberance is the olfactory bulb. And the olfactory bulb is the region of the bulb that actually receives the information coming from the sensory neurons. Sensory neurons actually project to that region. And here, and is the first part of the central nervous system that processes olfactory information. And you can see that in rodents is huge. It's like 1/5 of their brain is actually devoted to process sensory information, because rodents survive and reproduce to this sensory system as opposed to visual or auditory system as humans. So if we are going to use mice as our experimental model, it makes sense to actually use the olfactory system, which was the sense that they most engaged to actually survive and sense their environment. And the other nice feature about the olfactory system, and as you can see here, this is sagittal cut of the olfactory bulb, is that you can see how perfectly the layers can be observed. Oh, I'm sorry. How neatly organized the layers are. So you can immediately say that this is one layer. Here we have another layer. I'm going to explain the details just in a bit. And here we have another different layer. So we can immediately tell the circuitry. It kind of look like the hippocampus. I don't know if you have seen any histology of the hippocampus, but it's really structured so you can identify input and output really easily. So it makes a perfect system to actually study sensory coding and perception in mice. And here I have a diagram where I'm not going to give you all the details, but I want you to kind of focus on a couple of neurons that are going to be the neurons that I'm going to, part of the circuit that I'm going to be talking in my, in the presentation. So this diagram, odors here are entered the nose through the inhaled air. These are volatile chemical compounds. They interact with receptor olfactory receptors that are located in the cilia of the olfactory sensory neurons. Olfactory sensory neurons then project their axons to the olfactory bulb. And in all these little circles here that are also depicted here in this DAPI staining are called glomeruli. And in this glomeruli, the first synapse between sensory neurons and the projection neurons, the mitral cells, occurs. And then mitral cells project their axons to other brain regions, such as the piriform or olfactory cortex. So this is really interesting because if you know, the olfactory system is the only sense or sensory pathway that does not pass through thalamus. So it goes directly, it's only 2 synapses away from the piriform cortex. So there's a lot of sensory integration and codification also occurring early in this sensory pathway. And another interesting neuron is this one here that is called granule cells, which are the interneurons of the inhibitory neurons of the olfactory bulb. And they're massively located and expressed in the bulb. And you can see all of this layer, all this big chunk of thick layer, all of these are just inhibitory neurons. And the thin line here are excitatory neurons or mitral cells, which are going to project to the cortex. And it's only a little line. They're very orderly organized. So the question is, how does neuromodulatory or feedback actually affect sensory perception? Well, and this is a nice drawing by Santiago Ramón y Cajal that I really like. And here you can see the olfactory epithelium, the sensory neurons, the glomeruli, mitral cells, the excitatory neurons that project to the cortex. Here are the interneurons that I was talking to you about, granule cells, the inhibitory neurons. You can see that it's going to the cortex, but look at this part here. So he also kind of draw a lot of feedback afferents here. At this point, he didn't know what they were, but he actually got it right. So you can see this arrow and the direction of the arrow, it actually says that it's going towards the sensory region. And now we know that these fibers are neuromodulatory fibers or centrifugal feedback fibers, because the olfactory bulb receives a lot of feedback from either regions like the olfactory cortex that process sensory information by itself, olfactory information, but also from other neuromodulatory afferents, such as cholinergic fiber from the basal forebrain, noradrenergic fibers from the locus coeruleus, serotonin fibers from raphe, etc. And all of this massively innervates the olfactory bulb. So the question that I have was, well, what is the role that basal forebrain neurons play during olfactory discrimination? And why did I choose to focus on this particular region? It's because the basal forebrain is a nuclei that it's located here. This is a mouse brain, but it's also, of course, located in the human brain as well. You can find it. It's a nuclei. It's all this part here. And it's a region in the anterior basal forebrain that is composed of several nuclei. And this basal forebrain actually have neurons that project widely to the cortex, to olfactory bulb, to piriform cortex, amygdala, etc., to several brain regions. And there are a lot of brain function that has been linked to the activity or activity of the neurons that form these nuclei, such as arousal, attention, learning and memory, sensory discrimination, and it's the principal neuromodulatory system involved in neurodegenerative diseases. And this is specifically considering one type of cell, because the basal forebrain is very, very complex and heterogeneous. So the type of neurons that you can find here is very diverse. So you can find cholinergic neurons that are one of the most widely studied here in this basal forebrain, but you can also find glutamatergic projection neurons and GABAergic projection neurons, long-range projection neurons. To me, it really caught my attention to understand at one point what was the role that this long-range GABAergic projection neuron had to the olfactory bulb, because most of the research that had been done until the point that I did this study was that there was always excitatory feedback or neuromotory afferents, and but these GABAergic long projection neurons that are also kind of can shape sensory perception when kind of mislooked. So in 2010, not a while ago, not very far away, what happened was that there was this paper from Gracia Llanes that actually showed that a specific nucleus of the basal forebrain called the HDB, or the horizontal diagonal band of Broca, but I don't want to bore you about with the specific name, but a specific nuclei of the basal forebrain, the HDB, project to the olfactory bulb. So the olfactory bulb receives exclusively projection from this nuclei in the basal forebrain, and that there were GABAergic long projection neurons that actually reach the bulb from the HDB. So if you can inject a tracer in the HDB, days later, weeks later, you could do a sagittal section, like a histology in the bulb, and you can see all these fibers reaching the cranial cell layer, mitral cell layer, and glomerular layer. So after this, the question was like, okay, so now we have long-range projection neurons that are inhibitory. We have a lot of inhibition in the bulb. What is this GABAergic projection doing, and how are they affecting the sensory circuit, the local circuit in the olfactory bulb? And if we find that they do something, what role do they play in olfactory guided behaviors? So by now, I'm sure that you have, you know what channel rhodopsin is. So we took advantage of the genetic techniques because we wanted to, since the basal forebrain region is so heterogeneous, we wanted to selectively label GABAergic neurons with channel rhodopsin so that we can selectively stimulate these fibers and see what effect do they have in the local olfactory circuitry. So we had GAD65 Cree animals. GAD65 is located in most inhibitory neurons. And we made an injection in the basal forebrain with a flux virus with TD tomato that has the reported TD tomato, so that we could express, oh, I'm sorry, there's an error here. It's not why it feeds TD tomato here. So that inhibitory neurons from the basal forebrain could selectively express channel rhodopsin. And after we did that, we did the injection, we did brain slices, and we did wholesale in vitro wholesale patch clamp recordings from the olfactory bulb while we were flashing blue lights. So what we do is that every time that we flash light, we will stimulate the axons of these basal forebrain GABAergic neurons. We were going to release GABA, and we could see what effect that activation or GABA release from this long-range projection neuron had in the circuitry in the olfactory bulb. And this is how the animal looked like. So we did the injection in the basal forebrain here. This is, I think, 48 hours after the injection in the basal forebrain. And we waited 3 weeks or 4 weeks. And so that kind of rhodopsin could be expressed in the axonal terminals. We did brain slices, and this is how the olfactory bulb looked. So you can see that it's massively innervated by these long-range GABAergic fibers from the basal forebrain. You can see these, and these long-range mostly innervates cranial cell layers. So it innervates mostly the inhibitory neurons. So that was curious. So basically what happened was that inhibitory neurons were actually targeting inhibitory long-range projections were actually inhibiting the inhibitory neurons. So we wanted to see whether that was actually in fact true. And here we have a diagram. We have the glomerular layer of the olfactory bulb, the mitral cell layer, and here we have the granule cells. So we were doing whole cell patch clamp recording from the granule cells, which are the inhibitory neurons, while we were flashing light and activating GABA release from the terminals of the basal forebrain. And here we can find that when we actually applied light, the number of action potentials that were generated in granule cells were actually greatly diminished. Yeah. And this was the same thing that we did in another brain region, which is called the accessory olfactory bulb, that's actually in charge of processing pheromones, but I don't want to go into detail in that. So we did a lot of experiments here. We determined currents, and we actually determined that these inhibitory projection neurons were actually releasing GABA and inhibiting granule cells. We did pharmacology that I'm not going to show here now. But next then we said, okay, so now we have a local physiological role that these neurons from the basal forebrain are producing. So what is the behavioral relevance of the centrifugal inhibition? And to do this, we decided to do a very simple behavioral task that is called habituation dishabituation, and that is going to explain in a second. And we were going to have two different groups of animals. We will have control animals, but also animals that we were going to, through a viral injection, express DREADs. DREADs are mutated muscarinic receptors that are activated by an endogenous compound that cannot be found in the animal's body, that is called CNO. So through injections, you could actually, in genetics, you could selectively express this DREAD in GAT65 animals. So we had the GAT65 Cree animals, and we did a flux injection of this DREAD that was inhibitory, because there are also DREADs that are excitatory. So we chose to do to express an inhibitory DREAD. And we selectively express M4DI in basal forebrain GABAergic neurons. And after 4 weeks, what we did is that we did the behavioral task, and into our treated group, we also apply a CNO injection. And with the CNO injection that is applied systemically, then it's going to bind to this receptor, and the whole intracellular cascade is going to be activated, leading to neural inhibition. So what we were aiming to do was to actually inhibit the inhibition from the basal forebrain and see what behavioral effect that this inhibition had. Again, we injected DREAD in the basal forebrain of these Cree animals, selectively expressing this inhibitory associated receptor into GABAergic neurons. We waited for weeks, and then we applied CNO so that we could actually eliminating the activation of this neuron. And we then expose the animal to a behavioral test, and that is called habituation dishabituation. In this behavioral paradigm, what we do is that we have the animal, and we present the animal with different odors. So first we present the animals with just water so that it can get habituated to the presentation, with the human actually placing a plate and a culture plate in the cage. And then we present the animal 3 times with odor A, and then a 4th time with odor B. So what will happen is that mice are curious by nature, so that they are going to investigate whatever we're presenting. And we can measure the investigation time just by recording the behavior, and then we can kind of assess how much time they spend near the object. And so when we present order A, at the beginning, they're going to have a very high investigation time, and then it's going to habituate because the animal is going to recognize and form a memory associated with that order, and the curiosity investigation time will decrease. Then when we present order B, or C8 in this case, what would happen is that if the animal discriminate and perceive this odor as new, it would also increase again the investigation time. If the animal does not identify this new odor as actually a new odor, the investigation time will remain low. So with that way, we can actually test whether the animal is discriminating or not different odors. And what I'm showing here is actually the investigation time in the last presentation of C7 and what happened when I presented C8. Yeah, so order the other order. And in this case, we decided to present odors that were really similar, only differ in how many carbons they had. So we can have 7 carbons in length, or 8 carbon in length, or 6. We wanted to test how they could actually finally discriminate these odors that were structurally similar. And we can see here that an animal that is controlled, the animal habituates to C7, and then when we present the odor that had 8 carbons, the animal discriminates and increases the investigation time. However, when the animals were injected with CNO, the animal could no longer discriminate. Okay. 4 hours later, when actually CNO is metabolized and washed out of the body, the animal actually rescue. It wasn't rescue, but the animal could discriminate again between C7 and C8. And we did the same experiments now because these odors were really structurally similar. We knew that it was hard for the animals to discriminate these odors. So we said, okay, so what if we present the animal with odors that are not as similar in structure? And so we presented an odor that had 6 carbons versus an odor that has 8 carbons. And we noticed that CNO did not affect the discrimination of odors that were more dissimilar. So what we concluded is that GABA release from the basal forebrain was actually required for fine olfactory discrimination. And if you're curious, I can talk you into the details how the circuit is working and how signal and noise are actually being equilibrated. So if you want to know more about this, we can talk about at the end of the talk because it's really interesting. But I want to talk to you about another part of the basal forebrain, and so I want to have time to do that. So that's why I'm not going to stop here for a long time. So we knew that the basal forebrain then had an effect on olfactory perception. So the next question that I have, so what role do basal forebrain now play during an active olfactory discrimination task, meaning that it was an operant task where the animal had to actually associate a cue, an olfactory cue, with a reward. And to do this, what I did was that I implanted an array of tetraodes. I mean, this is an implant that has 4 tetraodes, and it's movable. And we could actually implant that tetrode here in the basal forebrain, and I wanted to see how these neurons were behaving, when were they recruited or not during this active olfactory discrimination task. So here is the device that we built, and here is a magnification of the tip, and you can see here that we have the 4 tetrodes. We also have here an optic fibers that I'm going to talk a little bit later, but this device allows us not only to record, but also to regulate neural activity through light delivery. So, and here is an animal that is implanted, and it's also attached to my amplifier in my head stage so that we can actually record the activity. And the behavioral task that we use was a go, no go behavioral task. Maybe you already heard about this, but basically the animal needs to learn to associate one cue with a reward and another cue with no reward. So what will happen eventually is that the animal learned the task and always respond to the rewarded order and stop behaving and just refrains from leaking in response to the unrewarded order. So the way we do this is that when the trial starts, the animal approach the operant chamber here, start leaking to a tube so that the trial can start. The order is sent, and then the animal needs to discriminate whether to keep leaking if it's the rewarded order or just refrain from leaking at that point. And that is what we evaluate. If the animal continue to leak in response to the rewarded order, then water is given at the end of the trial. And this is the olfactometer that we use, that it was designed by the Restrepo lab. And now I have one here in my lab in Chile. They were really kind, so we built one and I brought it with me. So now that's the animal here. So here we have the other tubes. We have here a pump that is actually flowing, pushing air. And everything is computerized, and you can actually change what orders you want to give the animal. And then you record leaking, trial starting time, and of course, correct and incorrect responses. So you have 4 types of responses here. You have 2 types of correct responses and 2 types of incorrect responses. Correct responses are the animal leaked in response to the reward order, so it got water. But also a correct response is when the animal refrained from leaking in response of the unrewarded order. So those 2, hit and correct rejection, are correct responses. Then you have 2 other possibilities where the animal, the rewarded order was sent, but the animal refrained from leaking. That's an incorrect response. Or when the animal received the unrewarded order and the animal kept leaking. So those are called false alarm and misses, and both of them are incorrect responses. And this is how it looked when the animal is implanted, and it's actually there, the animal is leaking. So the rewarded order was sent, the animal kept leaking, water was rewarded. Now the animal is going to move back, it's going to start again. And you can see that the unrewarded order was delivery, and the animal immediately retrieved and from the port. So usually the animals behave like this. Animals, this is a very, very easy task for the animals. So the pre-training takes about 1 week, and this is the first training where we actually test the animals and train them to discriminate between isoamyl acetate, which is banana odor, and mineral oil. Mineral oil is what we use to actually dilute the odors. So basically it's like the solvent and the odor. And they learn pretty quickly, just in 1 day, in 1 session, they can learn. So 1 session, we train the animals daily, 1 session per day, and 1 session, it's divided in 200 trials. And what we do here is that we average 20 trials, and we call that block. So we can see here that we have 10 blocks, which means 200 trials, and we average the behavioral performance. So here you can see that at the beginning, the animals start behaving at chance level, 50% of correct responses, what we would expect, right? And as the trials progress, the animals quickly learn that how to associate the odors and reaching criteria, which is over 85% the first day. Then the second day, we expose the second day just to make sure the animal learned the task, and the animal even faster, they reach criteria. This is interesting, but we're not going to talk about this here. Anyhow, and while the animal was doing this, then we're doing recording, and after the raw data was, of course, obtained, we had to do spike sorting and data analysis. So what did we find? So we found that something very interesting. Remember that we are recording from the basal forebrain when the animal is performing the task. And when we align all our units and firing rates to the T-start, so when the animal started the trial, we actually found that this is one unit, this is another unit. Each of this line is a trial. Each of this tick here is a spike. And you can see that these units in particular, here in the bottom might have the average, yeah, of all the trials here. We can see that this is T-start. T-start is when the animal actually enters the port and breaks a beam that I have here there on the machine, so we know that the animal actually entered. This is voluntary. There is no cue that tells the animal when to enter the port. The animals enter completely voluntary and out of their own motivation. So we see that milliseconds after the animal actually enters the trial, there is a peak of activity in the basal forebrain, either with units that are inhibiting or units that are getting activated. So when I did the population analysis, here we can see now each of these lines is the average response of one unit, and they're color coded. So the higher the firing frequency, the hotter the color or the whiter. The lower the fire frequency of that particular unit, the darker the color. So here we have 53 units out of 150 that we recorded in total. And these 53 units show a statistically significant change before and after T-start, which is the time period where the animal actually broke the beam. So you can see here that this is T-start or T-start. You can see neurons here that are getting activated, that are shown here with the orange sidebar, and a lot of neurons that are actually being inhibited, right? But when we looked closer at these time points and we see when the change starts, we notice that actually the changes started occurring in a lot of neurons way before the beam was broken. Anyhow, this was really interesting to us because that kind of told us that the basal forebrain was not only recruited for sensory discrimination when the order was actually being sent, but also that the basal forebrain neuron activity is regulated before the animal starts a trial, and even way before the olfactory cue was introduced, meaning that there is this anticipatory attention or maybe predictive coding. I don't know if you already talked about this, but this idea that the brain is constantly evaluating and anticipating to the environment. So it was really interesting to us to see this. And it makes sense because either GABAergic or cholinergic release from the basal forebrain could actually prime neurons prior to getting the olfactory cue, so that it can respond better and actually discriminate better. And we did also find that during trial start, the more neurons that were recruited during trial start had a positive correlation with correct responses. So if neurons are actually recruited during trial start in a higher number, that was correlated with a higher probability of actually responding correctly to the task. Well, we also find this, and this is something that we expected, and this is actually what we were looking for. That was our hypothesis, that these basal forebrain neurons were actually recruited during olfactory stimulation. So here is when the orders were sent, the unrewarded and the rewarded stimulus, and you can see that there are neurons that actually increase and decrease their activity in response to the neuron signaling, of course, and helping the animals to discriminate the odor. The same thing with reward. That was also something that we knew that the basal forebrain was participating during reward. And we also found that there were neurons in the basal forebrain that were recruited during reward delivery. So we found that a subset of basal forebrain neurons were recruited when the animal is exposed to the sensory stimulus and presented with their reward, suggesting that these neurons are required for olfactory discrimination. But now, then we wanted to see, okay, I just wanted to make sure that I have 3 more minutes, right? Yeah, okay. And with this,
Go ahead.
Yeah, Jose, okay. So, and with this, I'll conclude. So then we wanted to know what was the role of specifically of the cholinergic neurons in these nuclei and how it was affecting olfactory discrimination and decision making. So what we did is that the animals were not only recording from wild type animals, but we were recording from animals that in addition had channel rhodopsin selectively expressed in cholinergic neurons. So after the animal ended the task, what we did is that we apply a protocol of light so that we could evaluate out of the population of neurons that we recorded, which ones were actually responsive to light and cholinergic. But that's not as easy. Oh, well, so here is actually how the animal looked. This is an animal, this is the basal forebrain. This is a transgenic animal. This animal was not treated with any injection. It's animals that were crossed with transgenic animals that were produced by crossing 2 different animal strains. And this is basically what we found. So you have your recording, and if you apply light, you could see that in vivo, there's an increase in activity in this animal, increasing activity that does not happen in animals that just chat cree. This is of course, our control. So the first thing that I did also when I had this animal was to know, is there a change in behavior when I activate these basal forebrain neurons, just to see whether I could induce a change in behavior. And this is an animal that is just in a chamber, and I'm just applying light to see the behavioral output of this. So this is an animal that is just chilling in there. It's implanting. You can see this yellow cable here is the cable that is actually attached to a laser. And now when I start applying the pulses, the animal increased their sniffing frequency, which is really nice because as opposed to other sensory system, the animal can voluntarily increase the sampling of the stimuli, of the olfactory stimuli, by increasing the sniffing frequency. If the animal sniff at a higher frequency rate, then more odor is going to actually enter the nose. And but this was not too easy, because how did we know that actually what we were finding, the neurons that were responsive, were not, oh, there's something talking, so how did we know that actually these neurons that were responsive to light after the protocol and were responding to light pulses were actually directly activated by light, or were desynaptically activated by a cholinergic neuron nearby? So this is basically what happened here, whether I was recording from this one or from this one, but with the light I was activating neighboring cholinergic neurons. And as I show you, the local circuit of the basal forebrain is really complex. So this actually was a possibility. So in order to test this, in collaboration with Dr. Christian Sea del Rio, what we did was that we did brain slices of these neurons that were expressed acetylcholine specifically in cholinergic neurons. And we did wholesale patch clamp recordings there while we were flashing light. And we found that if you were recording directly from green cells, which were channelrhodopsin and cholinergic positive. And if you apply light, then the reliability of having an action potential was 100%. And the standard deviation of the timing was actually almost 0. And it was pretty quick. It was pretty quick. You gave the light, and of course, the response was really, really fast. As opposed as a neuron that was activated by neighboring cholinergic neurons, where action potentials in these neighboring cells were not only reliable, you have a latency, a longer latency. See here, the pulse was even, you have a longer latency of a response in action potential, and also you have a standard deviation in terms of timing that was not dependent on the stimulation. So after we calculated these parameters, we kind of apply, and we were really strict in applying those concepts into our in vivo data. And we found that from all the neurons that were here we have, so the latency is less than 10 milliseconds from the start of the pulse. So we make the cut there, and we found that like they were 20 putative channelrhodopsin neurons in vivo. Putative meaning that with low latency, but that could, when we apply all the criteria, we found only 6 out of all 153 neurons that we were recording from that were actually cholinergic. And that makes sense because cholinergic neurons, they have a huge impact in brain physiology, but only account to the 5% of the total population of neurons in the basal forebrain. So they have huge projection, but they're not that many. So it was not surprising to us that we only found 6. And to make a long story short, then we could see that they participated actually in this anticipatory attention period that we were saying during T-start, and also during olfactory discrimination when the order was actually set, but not in reward. So with that said, and now we're focusing on trying to understand what is happening in neuropathologies in my laboratory. And with that, I'm going to end the talk. And this is the acknowledgement section to, of course, my lab, Grupo de Fisiología Celular, my collaborators, Christian, Diego Restrepo, Molly, and Ricardo, and all the funding. So thank you.
Thank you very much, Alexia, for your excellent talk. Personally, I was very happy to hear it because I work in the olfactory bulb and during my PhD thesis. So I would say from all the talks, this is the one I understood the better because I have the anatomy and the physiology on my mind. We have a very short time for questions. We will give you the audience preference. Maria Triolo is asking, it has been observed that what is seen in the olfactory bulb is replicated in the olfactory neuroepithelium. If so, could this neuroepithelium, easier to obtain, be used to perform these studies? I think what she's asking, what happens in the neuroepithelium with this centrifugal stimulation for the forebrain?
Yeah, so it's a very good question. The thing is that it's actually really in the periphery, and there are cholinergic modulation happening in the neuroepithelium, and there are some techniques that can be used. For example, olfactograms that can be used in the olfactory epithelium to test whether there's actually a higher influence. But what I can say for sure is that olfactory sensory neurons project to the olfactory bulb, and in the first layer there is for sure neuromodulation of the olfactory, the acetylcholine in shaping how these axons release glutamate. That for sure happens. And actually, we do know that they can also shape their receptive fields of these glomeruli, and also participate in habituation, dishabituation at that level very, very early. So when an odor is actually being habituated, then if you could stimulate the release of acetylcholine in the glomeruli just there, the animal pay attention to the odor, and the animal start discriminating the odor again.
Thank you. We are a little beyond time, but I have to ask a question. This is something
Yes.
I like.
Sure.
You talk about predictive coding, and predictive coding also deals in some way with another concept that is active sensing. That,
Right.
as you said, the brain is not only waiting for the stimuli, but it's also preparing for being stimulated.
Mm-hmm.
So have you known or have you tested how this basal forebrain olfactory bulb interactions interacts again with the respiratory cycle? Because it's the way sensors act in olfactory. So you move, we say in visual field that you move your eyes.
Right.
And in
Right.
touch, you move your hands or whatever. But in smelling, basically you sniff. And that's the way you prepare for being stimulating and so on. So have you seen something about these interactions between basal forebrain and olfactory bulb during the respiratory cycle?
Yeah, so thank you for that question. It's really interesting because, yeah, so the frequency, as I was saying, sniffing frequency changes between a passive state and an exploratory state. Yeah, so when the animal actively sniff, the sniffing frequency increases even up to 12 hertz, which is really fast. So what I did was that actually was testing an animal in the sniffing cycle while I was applying light. And what I found was really interesting because there is a really, really neat correlation between light. I might have even the picture here, but I don't want to use more of your time. But every time I was applying light, the animal was increasing the sniffing rate. And that happened only in my channelrhodopsin animals, not in my control animals. But that is a side project that I didn't pursue longer because it's really hard to have, the fiber and this pressure sensor there in the nose. But it's something that it really interested me. Yeah, so it does. There is a strong correlation between cholinergic release and sniffing frequency.
Cool. We have another question, but I'm apologize to the audience, but we have to stop for a while. We have to take a break. Maybe you can send answers directly to Alexia. We can give you some contacts of her, but we now have to take a little break. So we will return at 11. We will came back for a technology showcase, but before that, once again, I would like to thank very much Alexia for her great talk. It's very exciting. I was very happy personally to hear about olfactory system, that it's a fascinating system. Thank you very much, all the audience. See you at 11. Thank you, Alexia.
Okay, thank you. Thank you for inviting me. Bye-bye.
So thank you once again for being here. So far, it's been a great symposium. Few reminders. Please, two things to remember. One, we're going live on YouTube. So all these talks and all the previous talks will be stored in the Universidad de Santiago Internacional YouTube channel, so you can watch them offline or not on real time if you want to. Second, please please remember to do all the questions through the preguntas y respuestas or Q&A button in the bottom of your Zoom screen. Please do not make it through the chat. The chat is for not for the question, just to ask us other things. And now we have a different idea or a different talk. This is a technology showcase. Most of the things you've seen here are done through or need a kind of complicated data analysis. There are many solutions for doing that. One of the most used in the scientific community is MATLAB, which is a software made by a company called MathWorks. So MathWorks is going to talk about technological solutions from analyzing neurobiology data. I'm a little lost here. Who will talk? Akhilesh?
Yes, sir, that's right. I'm sorry, I was trying to mute myself. This is me, Akhilesh, over here, and I would be presenting this talk.
Okay, thank you, Akhilesh. Thank you very much for being here. For us, it's also an honor to have you here. We as neuroscientists know the extended use of your products in neuroscience, and it's very it's a happy occasion to have you here explaining how the people who made the magic do the magic. Okay, so the screen is all yours.
Thank you very much. Just get all right. Let me start sharing my screen. Thanks for this wonderful introduction. And let's see if you can you confirm if my screen is visible right now?
I'm not watching it.
No.
Not yet. Okay, let me just.
I think now we are watching your screen.
Okay. So I have multiple monitors. Are you able to see my slides, or is it something else?
Neuroscience signal processing made easy. That's the one.
Perfect. Excellent. Thank you for this wonderful introduction. Really appreciate it. And like you already mentioned, so I'll be kind of walking us today with a little bit of techniques in MATLAB on how you can make this neuroscience signal processing a lot more easier, and kind of demonstrate some of the capabilities which a lot of the neuroscientists worldwide use as of today from MATLAB. And just to give a little background about myself, and I am representing MathWorks today. I'm a senior application engineer, and I help customers especially in the medical industry, in adapting MATLAB for wide variety of workflows, from signal processing, machine learning, deep learning, AI, and whatnot. I have been with the company for 5 years now, and working in this domain. Before joining MathWorks, I was working on some radar signal processing back in the day. For almost another 5 years, we were sounding the glaciers in Greenland and Antarctica and doing signal processing to understand, build the images of the glaciers up there. But it's kind of very equivalent to, one of the things which I tell to my medical customers is like I was doing the ultrasound of the earth essentially, or the ultrasound of the ice sheets, because the signal processing is kind of pretty much the same. But anyways, So just wanted to give, this is a few references from our customers worldwide. They use, MATLAB has been extensively used for all these signal processing tasks in medical, and a few interesting tasks which have been recently, like these are some stories which customers use MATLAB, they're successful, and then they share it to the public. So here's one story where Battelle Institute, what they did was for they used MATLAB and Simulink for a paralyzed arm hand movement restoration. So using the signals from the brain, they are able to decode the corresponding movement and then have all these sensors on the arms which kind of stimulate the hand movement. So this was a very interesting work which they did using MATLAB and directly collecting the signals from the brain. You can see that this is one sensor on top over here. Here's another good research done at UT Austin, where they acquired the MEG data from the brain and used MATLAB to convert those MEG data in some signal processing, what we call as time frequency representation, which is also called like scaleogram, and then use deep learning networks to actually decode that signals and into speech. So, for ALS patients and whatnot. So this is a very groundbreaking research which they had done, and they have used our tools to do that. So what we'll do today in the next 30 minutes or so, we will actually see some of the capabilities of MATLAB at a high level, what I think you guys, the audience today, can leverage from. And we'll work on an example on training a EMG classifier. So we have some signal data from EMG with the different movements of the hand. So there are like 8 sensors which are acquiring this signal from a patient. And what we'll see is we'll explore a lot of the signal processing capabilities on extracting features and extracting something meaningful from the data. And then also train a machine learning classification. I won't go into too much details in the interest of time and also from a value, but because machine learning is a big topic in itself, so we can spend almost 1 hour on that exclusively, but we'll just quickly show you how easy it is to perform all the signal processing tasks and even train a classifier such that, like, once we get a new EMG signals, we are able to use that classifier to automatically tell what grip what hand movement is in that signal. So kind of walk you through that process. So with that, let me switch my slides and I'll jump to MATLAB. And if there are any questions anytime, please feel free to continue asking it as I'm presenting. I'll be happy to take the questions as we go along. So let me just quickly share my screen here. So hopefully you're able to see MATLAB open. And what I'll do is,
we're seeing MATLAB.
Perfect, very nice. So what I'll do is, I have this one script open, but you will see, like, although this code is there and we probably can share the code afterwards as well, but what you'll see here is like all the data processing and everything I do, I would write like minimum code, and I would just still be able to explore signal, analyze signals, extract features, and even train it with the minimum amount of coding. So let me quickly show you what that data looks like. So by the way, this is for those of you who have never seen MATLAB, this is our scripting language. This is what we call it as live script. And what this live script allows you to do is like insert images, equations, and text along with your code if you have any. So this live script kind of is a very interactive feature on making you work with some code, realizing displaying the results and whatnot. So I have some data which is sampled at a rate of 1800 hertz. And let me just run this section. So in this one, I'm just loading all the data. So the data is in my data folder, which looks something like this. So these are all, if you notice here, these are all CSV files. And let's just open one of these CSV files. Let's say for chuck grip, I have this data in Excel, which looks something like this sort. So this is data from 8 sensors. With different locations of the arm, and then the last column I have is my classification or the action for all these different EMG readings. So what I'll do is in MATLAB, it's quite easy to load these signals, and that's what I'm doing over here. Let me just go back to my path. I just create a data store by giving the path and calling that, okay, in this data folder, whatever is my training data, that is what I'm going to load. And you can preview it, but then you can call this read all command and it will just read all the CSV files automatically. Let me just run this section. I haven't run this. So you can preview it, and read all command reads in all the data. And now the data is in MATLAB, and let's see the data again. So now you see that in MATLAB, it created a table which was similar to the CSV file with all these sensors and then with a corresponding action. So in this action, if I kind of scroll down, so what I'll see it, I actually not only did load one CSV file, but kind of like collated all the CSV files so that I have all the actions. So key grip, no movement. We have different classes. Let's see, let's just scroll down a little bit more. So there's other classes of the wrist movement, such as, abduction, wrist abduction, and then pronation, supination, and whatnot. So we have all these signals all there. So now there is a lot of code below, but this is for reference, and I'll tell you, what it really means just in a bit. But just to give an idea of one data, like if I were to just plot one of those signals, so this is how my table looks like. So if I read like one column from the table, and so these are the different categories, and with time, the person whose data we have collected, he's going to change his grips. So, start with the chuck grip, then with a fine pinch. So this is just one of the 8 sensors. But if you'd like to see like all the 8 sensors, this is the code here, and this is just for the visualization purposes. But let's just, let's say like if you want to see all the sensors plots, then this is what it looks like for all sensors. So this is like 8 sensors for one of the actions, which is like key grip in this case, right? So different plotting tools are available. Like in this case, I'm doing a stacked plot. In the previous one, I was doing a regular plot, which we call a scatter plot. But again, this is just for visualization purposes. But now let me open another script and let's go into the analysis. So whatever I'm going to do in the next steps is actually taken from this research paper, which I got, and build this content off. So this is some Motion Labs an article on EMG analysis, and they also kind of walk us through the process on what is EMG and how do we do the signal processing of the EMG, and especially if I have to build a classification model, then what are the things I need to do to build this EMG. So I'll just walk you through that process, but using one signal initially, and then we'll go and look at other signals, how we can automate this feature extraction process. So if I go back to the slide, essentially it would be just real quick on the slides. The signal processing is a step where I'm essentially doing a feature extraction. So using these signals, I can actually try to train a machine learning algorithm, which I'll see, but directly from the signals, the algorithm is not so powerful. But we will do some feature extraction is essentially with signal processing to actually build a classifier which gives me some good results. So let's take a look at it. So when I said I can build a feature, a classification from directly from the signals, this is how I can do that. It's very simple. In MATLAB, what we have provided these days to, for facilitating and easing the task of classification, learning, signal feature extraction, and whatnot, we've provided all these GUI-based apps with different areas. Let's say you're working in signal processing, we have all these apps for signal processing. If you were to do deep learning, machine learning, we have these apps for that task. And let's just open one of the apps, which is called classification app right now. So it opens this interface, and this is a classification learner app. As the name suggests, it will train some models to build all these classification algorithms. So what I can do is, let's say I don't want to do signal processing initially, and I want to see on the raw data how it looks like. So I can just quickly start a new session, select input, and right now my input is trained data. It recognizes tables. And if you see over here, what it did is automatically it figured out that, okay, the last column is action, and which is like 15 different categories. and everything else is data. So all this 8 sensors are classified as predictors is the data from the 8 sensors, and this is my action. And I can save like validation and whatnot, but let's not get there right now. So if I start a session, you'll see that, if I'm new to deep learning, machine learning, I don't know which algorithm to train. One of the features which we provide is you don't have to know the algorithms. In this drop-down menu, we've provided you all the algorithms over here, like medium KNN, coarse KNN, and whatnot. But if you do not know which algorithm would work better versus the earth, you can just also select like all algorithms and train all of them. So very quickly, what I'll do is I'll just select a few algorithms which are quick to train and just click train. So what this machine learning app will do is it will, now that it has loaded all this data of 8 sensors, and you can see that we have this color combination for all the different classes or the actions, because the data is a huge signal with the different actions at this point. So it will start training all these different models, machine learning models, and give me like an accuracy. So training, doing some learning and telling me like if it's good or not. So like in this case, it started like, the quick algorithms like tree, KNN, fine KNN, and whatnot. And obviously I can train all algorithms, but in the interest of time, it takes a while to do all these trainings, so I won't go there. But if you see like my results right now, they've started to appear, and my accuracy is very, very poor. It's only 28%, 18%, 13% with these algorithms. So obviously, directly if I feed the raw data, I'm not going to get any good results, and I'll just stop the training. So raw data with all the sensors is going to give me very, very poor results. And if I see a confusion matrix on actual class versus classified, you'll see that it's erroneous. This is not going to work. So that's why signal processing is important before we do this classification. So let's go ahead and take a look at that. So what I'll do is I'll just extract one signal from this trained data. And a good thing with MATLAB is as I'm typing, it is auto completing my code. So let's say I wanted to extract one signal and I'll save it as X. So in trained data, if I put moment I put a dot, it asks me, okay, these are the different columns where you can choose. And then let's say I just want to take first sensor. So I'll just go ahead and run this. I'll just do run section just X. And now you notice that this X variable has been created. And if I plot this, I can go to this plot and also just inspect it visually. So this is what the plot of this first signal looks like. And different regions are the 15 different classes. So now to extract features, again, without writing any code, I'll go to another app, which is called Signal Analyzer app. So if you look over here in signal processing communication, you'll see a lot of apps, but the one which I'm going to use is Signal Analyzer. And now following the steps of that document, which I had shown you, I'm going to do everything on this app itself. So this is my signal. It automatically recognizes that I've extracted one signal. Let's go ahead and pull the signal in the app and plot it. So this is what the signals look like. I mean, the EMG data with different regions and whatnot. Now, the first step I need to do is I have the code here based on everything what they have done. So the first thing which they recommended is to remove the DC offset. So let's go ahead and take a look at that. How do we do it? So obviously, if you see just by one signal sample, the DC is around 4.40,000, and the signal is varying around it. So first thing I need to do is normalize all the data. So there are various ways to go about it, but here in this app itself, we have given you a lot of functions to leverage upon and reuse. So here's one of the functions is like detrend. So I can just quickly select this detrend function, and this is my signal X. So what I can do is I can create a copy of it so that I don't lose my data. So this is like same signal X. And what I'll do is on the copy, I'll just say that I want to detrend this signal. So eliminate any trends appearing on it. So let's go ahead and click play on it. And now if you see my original signal and this plotted, it automatically removed the DC. So very simple thing to do, but still without writing any code, you can kind of leverage it. So this is one thing which I did on one signal, and my goal eventually is to do on other signal. So one of the things which MATLAB allows you to do is anything you do over here on this signal, you also have this automatic code generation button. So you can generate a function from it and it writes the function. So this was a simple operation. So it created this function pre-process and automatically generated the equivalent MATLAB code for it. And we'll use this later on. So this is the first step. So now that our DC has been removed, let's work off of this signal. So let's go to the next step. Now, the next step is that, okay, we need to apply a full wave rectification. So how do we do that? So for those of you, you might be familiar with rectification, but if not with the rectification, I'll just point. So what we really need to do is rectifying means like you eliminate all the negative values and just retain all the positive values. So how do we do that? In the analyzer, again, you can take a look at this different options. You can see that there's filter, smoothing and whatnot, but there's not really any function which tells you like my rectification process. So maybe this function is not there, but what we can do is we can actually go in Google and also search like how to rectify a signal in MATLAB. And let's see what we get with it. So you'll see like there's lots and information of it. And maybe it's, maybe you already know off the bat, but here is like one MATLAB Central, which is a good resource to search things which we probably, if we do not know. I mean, rectification is a very simple process, but just I wanted to show you the workflow on how to search it. So here is somebody, actually one of our staff, he's telling like, hey, in MATLAB, you can use this function absolute to do that rectification. So what you can do is now just find this function absolute MATLAB, and you can see that, okay, absolute value of a signal. So this is the format is what you want to use. So ABS is the call, as simple as it is. So now if you want to do it in an analyzer, you can actually, this allows you to add some custom functions if some function is not available. And I'll just call my function as, my rectify or something. I will skip the description for now. And it says it does not exist. Do you want to create it? Yes, I can create it. And it gives me the template on what would fit in that app. And now I'll just say, I won't touch anything else. I'll just say that, okay, on the input signal X, I just want to apply this function. And I'll go to this and just save it. So absolute. So this is one of the workflows you can add custom functions. And now you see that my rectify function is there. And if I click this, and let's say I will duplicate this again, just so that I don't lose a copy. And on this one, the duplicate one, if I do the pre-process, so it created a function my rectify, and there we go. All the negative values have been removed, and all I have is everything in positive. So it did apply that rectification purpose. And again, for this task, you can, although it's a custom function, but you can again generate the same function, and you'll see that it creates this my rectify. And right now, my rectify is a very simple one, but you can have like a series of operations inside it, like one after the other. So it can be anything. Literally, it can be a very complex code. All right. I'll just take a pause for any questions at this time. I don't see any questions on the chat or Q&As. I'm trying to watch as I'm presenting.
Achilles,
yes, sir.
We instructed the audience to make the question at the end of the presentation.
Oh, okay. Okay, perfect. Then I'll open the floor for questions towards the end. My apologies. Thank you very much for letting me know. All right, so now that we have rectified the signal, now let's go to the next step. So the next step is that document, again, it suggested that we need to find the envelope of the signal. So envelope of the signal means like, okay, this still has a lot of variation. So this is not a good representation. Let's try to build an envelope around it. So different ways to go about this envelope creation. And let's go ahead and see this. Again, going to this pre-processing step. So one of the ways is low pass filtering. Let me try that. And if you see in this low pass filtering right now, in this options, it gives me a passband frequency. But actually in my data, I already have a sample frequency. So what I can do is I don't have to worry about figuring out what this, let's say I don't have a signal processing background. I just know that I need a cutoff frequency of, let's say, 15 hertz or 20 hertz. So I don't have to worry about all this radian samples, whatnot. But what I can do is in the signal, I can add time values, and I can tell it that, hey, I already know the sample rate of my signals. So my sample rate initially, what I loaded was FS, which was 1800 hertz. So I'll just put in 1800 right now. And now you'll see that my axis has turned into the time axis because I've given the sample rate. And even in my low pass filter, now I don't have to think anything. I can just specify my frequency. So let's say with a low pass filter, I want to have a passband frequency of 15 hertz and an attenuation of 80 dB. So anything beyond 50 hertz should be suppressed by 80 dBs. Let's go ahead and low pass filter it. So, all right, we do see some envelope coming up, but it may or may not be the best envelope what we're looking for. So this allows us to kind of quickly, let's say I reduce my passband frequency and see the results. Maybe this is also not the best. So what I can also do is undo this pre-processing if I don't like it, and I can retrieve my original signal. So I'm like, okay, low pass filtering is not giving me a good envelope. So what else can I do? So I just undid all that process, went to this analyzer app, and then there's actually a function which is called envelope. So let me try that. So in this envelope method, let's say that, okay, I'll just use a envelope, like an upper envelope, because I want the envelope on the top, not on the lower end, because the lower end is already zeros for me. And I'll use, let's say, RMS filter with some number of samples being, let's say, 500. And let's try to create this envelope. All right, awesome. This is exactly what I want, for instance. So you can kind of quickly go around by changing the settings, and let's say this is exactly what I needed, the overall envelope. But if this is too much, I can again do the undo pre-processing and create the envelope again and whatnot. So kind of go iteratively. And once I have a good understanding, I can again generate the code for everything I'm doing till now. So in this case, this is actually an envelope function with the window length and whatnot. All right. So now that I have an envelope, now let's go to the next step. Is okay. Now the low pass filter envelope did not work, but this worked. And then the next step tells me that, okay, now let's see the frequency features. Now for frequency features, let's calculate some of those for the different classes. Now remember, now that I've got this envelope, but this I still have it for all the classes. So let's go ahead and see the frequency spectrum of it. So now I can go to this tab, and now this tab, display tabs, gives me all these options of time, frequency, spectrum, and spectrum. But note that the signal length is for the entire signal, and this is not for my one class. So what I'll do is, let me actually reset this for samples again, and I'll tell you why I'm doing it. And I can go back to my data train and see till what sample length is my class number 1. So if I scroll down, I see that my chuck grip sample is all the way till 1 to, let's take a look. And I can do all this in code. I don't have to do this, but I'm just showing you a simple process. So 1 to 5130 samples is my chuck grip. So what I can do here is, I have this option of panner, and this is another tool which quickly allows me to observe a certain section. Now what I can do is, now I can either scroll or I can just give in the samples. So it's 151, what was that? We'll go back again, 5130. I can just give the value time limit 5130. So it's out of the whole signal, it extracted that 5130 part, 1 to 5130 samples. And I can just say that, okay, extract the signals between the time limits. Now it created another copy of the signals. And this one is just one sample to 5130. The original signal was the full signal, all the way from here to entire, but now this extracted signal is just that part which is called, which is for the chuck grip. And I can just rename this like X_chuck grip. right? And now since I have the signal of one class, I can just quickly plot the spectrum and get some inference out of it. So this is what the spectrum looks like. And if I do not like the spectrum, I have to change some settings. Let's say my overlapping window, I can quickly make those changes and observe how the spectrum actually changes. So you can see that how the spectrum is changing and whatnot. And again, for the spectrum task, once I have what I need, I can again generate a script. Not only can I generate a script for spectrum, but I can also generate a script to calculate the region of interest between time limits. So let me just create a script for spectrum. So everything I did with the original signal, I'm taking this, calling this function PS spectrum. It automatically created my frequency limits and everything I'm doing on the app essentially. So basically now at this point, when I've taken the spectrum, I am ready to train my classification learner app. So again, just to recap the things I did, I removed the DC from the original signal. So this was my original signal. Let me just remove the spectrum. First of all, I removed the DC. Then I did envelope extraction, which is my purple signal. And finally, what I did was I extracted certain regions like a chuck grip, and I'll do it for other classes too, and then create a spectrum of it. And these are the features which I'm going to use to train my model. So now that I have all this code for all these steps, what I have done over here is, if I go back to my script, I created this function. Let me just reopen it. I created this function, generate signal features, and essentially just use that code which was getting generated. I added another one like a variance, which I did not discuss, but it's just like simpler to absolute. I'm just taking a variance, and then I'm doing all this detrend and filter and whatnot and PX spectrum to it, and I'm just creating this feature set. So I've put all these things which I did on the app and all this code which was getting generated as one function. And what I do is I just call this function right now. So there's this block size map. Let me just go back up. So this is the part of the code where I'm actually, so that 5130 step where I calculated like, okay, signal 1 to 5130 is chuck grip. I can do it automatically using this 2 lines of command. So what I'm doing is this function mode, and I'm doing a count categories. So I'm counting the categories and figuring out the different modes. So this is my n action, and you see that, okay, it figured out that, okay, 5130 in this whole data set is actually what corresponds to sample size of one category. And this block size is what I'm calculating is how I need to divide my data for each category. So I'm automatically kind of figuring out those mechanisms, and this is what I use in this function when I call this generate signal features. And what it would do is I'm using this function again, block proc, to call this function because just to speed up the calculation. So if I go and help on this document, you'll see that we'll again come to the document. I can also do a Google search on it, but it does a block processing, and essentially under the hood, it is like got a parallel support. So with a parallel computing toolbox, so with 4 cores on my machine, I'm doing it quickly for all the signals. So because now I have to do that for all the signals, and that's why I want to do quickly on parallel. So like over here, you can see that it is processing that 1350 samples for each. So it divided that 5130 in the respective samples and created this another table, which I called as from the train data, which I called train data SP. Again, it is still all these actions, chuck grip, but now instead of 8 sensors, it extracted these features out of 8 sensors, which is the variance, envelope, the power spectrum, and for sensor 1, variance, envelope, power spectrum for sensor 2, variance, envelope, power spectrum, sensor 3, and so forth. So that's why 8 times 3, which is 24 features. So 3 features for each spectrum is actually which I extracted from signal processing and corresponding action. So converted this train data of this size into this train data SP of particular this size. And now let's go back to our signal analyzer, classification learner app. I'll start a new session again. And this is like the moment of truth on how if this signal processing actually helped or not. So let me see where this is getting open. This is a problem when we work with 2-3 windows. Sometimes the pop-ups which are coming, they don't appear. Just bear with me. Okay, there we go. So this started with a new session, and this time I'll give this train data SP as my input. So which is of 25 columns because 24 features and one last column is action. And now I'll start this session again. And again, now this data distribution. So it's still all that same data categories. You can plot now different features, like if I have to see variance of 5 versus variance of 3 for the different categories, you can see the scatter plot distributions. But quickly, I'll do the same thing, all quick to train algorithms without choosing which algorithm I want and start training it. So let's take a look at the results now. And now, as you can see, like even for simple algorithms like tree, earlier I was getting 28% accuracy. Now my accuracy is drastically improved, which is 85% accuracy. And these are just one of the few algorithms. So actually my model is learning. But if I go back to my script again, and if you see let's see if I can, there's a lot of algorithms. And I think the algorithm in which I got the maximum accuracy was probably, let me check. It's probably like one of the KNNs or something. Let me try one more. So let's say that, okay, course KNN, let's give that a so we can, quickly, actually, it's so simple to load any algorithm and start running with it. And if you want to change some hyperparameters, you can change it. So in the interest of, like, usually I'll run all the algorithms, but that obviously takes quite a while. So it's not advisable to do it. I mean, for your case, it is advisable to do it, but right now it's not feasible to do it in while showing you the demo. But I'll just cancel all these. I don't want them to run because some of the algorithms are more complex and they take a while to actually get some results out. So over here, this is the winner now, quadratic discriminant. And you can see also plot the confusion chart and you see that, okay, this is actually performing really well, only a few misclassifications and a 92% accuracy. So if this algorithm is what you want, you can, again, just like other apps, generate a function. So you can generate a function on how this is training. So it creates all this code. So everything we did in the app, again, it is creating all this code over here. And you can save this function. So this is exactly what I've done over here. But also at the same time, if you go back to the app, you can also export the model if in your workspace if you would like. But really now what we have come down to is, if I go back to the script, what I did was I actually created this function which was auto-generated by this app. So this was the function which I was showing, train SP classifier. And it automatically does the training, and the winner, whichever the winner algorithm is, it creates the training routine for it. And I can, so this was the one which, so it got 93% accuracy. And I can have this another data set, which is a test data set, which this algorithm has never seen before, run this section quickly, and actually for the new data set, so the test data set which I'm loading, I'm doing it the same format. I'm calling the same function, which is my block processing to extract even from the new data set, the signals which the model has never seen before. I'm extracting the signal processing features like I did using this same function. And all I do is I call the predict command on the classifier model. So the classifier which has been created, I forgot to do that step. Let me just do that real quick. It will give me an error. I can export this model, like the winner model, and tell that, okay, save it as a certain name, which is a classifier. And I'll just give it a while. Perfect. So like I had expected, I'll get an error, but I'll export this model and let's name this model as classifier. Okay, so now this created this classifier over here, and let us actually, let me just run this. I'll just train this function one more time so that we don't run into issues. But if you see this test data in the meantime, which I created is a table, again, a table with some more data, which the network, and this data is again 8 sensors, same format, but this has never been seen before by the model. And this is like the actual class which we know. This is the actual class, and we'll actually call that predict function and see what classes it predicts. So let's go back here again to the script and make this prediction using this line of command, which is predict on the classifier. And now it gave me some prediction action. So it gave me, for all those signals, it gave me this categories of what it thinks that is and the corresponding scores of the categories. And we can now plot this confusion chart and see on the test data actually what was the accuracy. So if you see over here, so this is what it looks like on the new data, and you can see that, okay, it is classifying and doing a good job or not. So anyways, so just to summarize again, we're kind of approaching the end of the time, but you saw that how easy it was to work, first of all, load data from CSV files in MATLAB. Using Signal Analyzer, I, in one place, ran so many commands and visualized the results immediately then and there. Exported out all the code automatically and regrouped it in another, which I called a signal feature extraction. And then once I extract the signal features for all signals very easily on this classification learner app, I trained all the machine learning algorithms together to figure out which is the best accuracy. So that being said, going back to my slides again, real quick, one thing which I do not cover today is addition on top of that. You can also, if you're building a medical device, you can also, everything which I did, you can auto-generate a C, C++, or even a GPU CUDA code from it, and actually deploy your algorithm, machine learning algorithm, on these embedded devices like ARM Cortex or NVIDIA Jetson or even an FPGA. And you can have this algorithm kind of taking in the data, doing all the signal processing, calling this classification function and doing the classification, and build an embedded device such as this one, from your in one platform, which is MATLAB directly. So that being said, I would like to wind up at this point and take questions. There is a way to get started. So if you want to get started, there are a lot of free courses already available online. And you can see on the MathWorks website. So we have a free course for signal processing, free course for MATLAB, free course for machine learning, deep learning, and other topics. There's some more focused courses which are instructor-led and online. So on these various topics. So there's like this link on MathWorks training, which you can take a look at and go to this various trainings. So that being said, my name was Akhilesh. Please feel free to connect me on LinkedIn. I have provided my contact information over here. And your account manager in South America is Annette, my colleague. She couldn't make it today, but I've given her contact information too. If you have any further questions or follow-ups, or you would like to reach out to us for licensing or things of that sort, please do not hesitate to reach out. So with that, I'll stop sharing my screen at this point, and I'll open the floor to questions for the next 5 minutes.
Thank you very much, Akhilesh. Awesome talk. I have to say, if I have seen this talk before my PhD, it would have lasted a few years less. But it's amazing how things change. Everybody is doing the data machine learning and stuff in data, and you show us that, and now it seems very easy to do that in MATLAB. One question: what, for the people who doesn't know, MATLAB has a core program or software which uses different toolbox for different applications. There are tools for neuroscience or signal processing, and there are tools for economic people who works with economic, people who works with imaging, people who works with airplane or design and stuff. For all the things you have shown us here, what extra toolbox do you need?
Great question. So everything which I showed today, we just need 2 toolbox in addition with MATLAB, which is one is the signal processing toolbox for doing all the signal processing tasks. And then the second toolbox is the machine learning toolbox for doing the machine learning.
Okay, cool. One other thing that I haven't knew MATLAB has that is this live script. When did you implement it? At what point or on what version?
Great question. So MATLAB live scripts was introduced in 2017A. So which is like 4 years from now. So now it's yeah, I think scripts is very powerful, and it's, I think a lot of us might be using already open source. So Jupyter Notebook is also following the same format if you have already seen it. So MATLAB live script is a more advanced version of that Jupyter.
Okay, one last question from the audience is Nicole Juanchuto is asking, I'm interested in simulation of models of neural circuit. What kinds of tools I can access to do that?
Great question, Nicole. So by neural circuit, if you mean neural networks, then we have this toolbox, which was actually called a neural network toolbox, but now it's called a deep learning toolbox, which has all the capabilities of neural networks. And just additional information, neural network toolbox has been there with MathWorks since I think 1990s earlier, even before the deep learning became so popular. So this is a very mature tool at our end. And yeah, feel free to reach out to Annette if you would like to request a trial or would like to try that out. I'm sure Annette can help you get an access with that toolbox.
Okay. Any further questions?
Jose?
Yes.
Francisco here. Yeah. Yeah, I want to chime in regarding the last question. So I think she's referring more to like simulation of like actual like brain neural networks.
I see.
So in that case, there is a free toolbox that a couple of friends put together that is called Dynasim, that it runs in MATLAB, and I can send the link through the chat. So that one runs in MATLAB already, and you can simulate like like real neural networks in it with all kind of different parameters. Yeah.
One thing I will ask, I will like to add to that is that the scientific community has developed a very precise tool for different neuroscientific requirements. We have, for example, EEG Lab, which is developed for analyzing EEG signals. Field Trip, which is also a tool for signal analyzing in a neural paradigm. So one of the advantage of using MATLAB is that you have what MATLAB gives you, that is a lot, but if you want something more specific, you can go to the internet and see, because there are a lot of toolboxes developed by mainly scientists who address precise questions or paradigms. So that's another thing that's good for MATLAB. You don't have only answers in MathWorks, you have in top of that, or adding to that, you have answers from scientists who had basically the same problems you have. Once more, thank you, Achilles. I will use your talk just to remind that two things about next week we have a workshop with the hands-on session, and we will use MATLAB as a main platform. But we will go way beyond. We will start, this is a workshop for basic introduction in signal processing. So everyone, two things about that. One is that we had a lot of requirements, and we have only capability to do 25 students. So we already closed inscriptions. We cannot handle more inscriptions. So thank for all the people who were interested. We will make you know tomorrow afternoon if you were selected or not for the workshop. Once again, thank you very much, Akinesh. We will move on the program. Thank you and MathWorks for this amazing talk. It kind of personally I get surprised on how fast things are and how simple are. passing through days of coding to just pressing button is what we all expect in language. So thank you again. That being said, I need to ask you to please stop your video, because we can move to our next speaker. Our next speaker is Professor Maria Inés Berens from University of Chile. Just as a small introduction, Maria Inés is a MD and PhD from Pontificia Universidad Católica. He did a postdoc in Washington University, St. Louis, specializing in demences. And she's also a professor of the Department of Neurology and Neuroscience, Neurology and Neurosurgery, please, my bad, and the Department of Neuroscience at the University of Chile. She is also the director of the CICA, which means the Centro para Investigación Clínica Avanzada at Hospital Clínico Universidad de Chile. For us and the people who know her, it's an honor to have her here. Lizzie, it's an amazing person and a wonderful scientist. So, Lizzie, thank you very much for accepting our invitation, and the screen is all yours.
Thank you, Pepe. It's a pleasure for me sharing with you about Alzheimer's disease and memory. And I also asked the organizers, Antonello and everything. My name is Maria Isabel, but everybody calls me Lizzie. That's why Pepe made a mistake.
Oh, sorry, Lizzie.
But that's okay, never mind.
Lizzie, one question. Would you like to speak in English or in Spanish? Feel free to.
I will switch to Spanish. I prefer talking in my own language for the presentation.
Yeah, thank you, and please forgive me for my mistakes. I'm a little bit nervous this time.
Okay, mi tema es el mecanismo del deterioro de la memoria en la enfermedad de Alzheimer. Es lo que me pidieron que les contara. El Alzheimer es una enfermedad degenerativa. No sé cuánto saben ustedes de ella. Mucha gente sabe mucho porque tiene familiares o porque lo ha leído en las noticias, etc. Yo voy a tratar de ser lo más general y lo más específica que se pueda. Y se sabe que destruyen irreversiblemente varias funciones cognitivas, pero la principal y la inicial en el Alzheimer típico es la memoria. Es una de las formas de demencia, es la más frecuente de todas, se cree que más de 35 millones de personas la sufren, y desgraciadamente aún no tiene una cura, aunque esperamos que pronto aparezca alguna eficiente. Como les decía, demencia es el término genérico que engloba todos los deterioros de la capacidad cognitiva de los individuos por sobre lo que era para ellos anteriormente y que compromete las actividades de la vida diaria. Eso se define demencia. Y dentro de ellas, la más frecuente es la enfermedad de Alzheimer, más del 60%. Después están las otras demencias como la demencia vascular, la demencia con cuerpos de Lewy, la frontotemporal, otras demencias. Y es muy frecuente que se combinen. Y si uno mira el cerebro de un individuo que murió después de los 80 años, es frecuente que tenga patología de Alzheimer, patología vascular asociada, Lewy asociado, etc. Es menos frecuente que la frontotemporal se asocie con Alzheimer, pero en general es muy frecuente que haya una patología combinada. La enfermedad de Alzheimer la describió Alois Alzheimer en 1906 en una paciente llamada Augusta en Alemania, y él describió estas placas seniles, se llamaron, o él las llamó, o no sé si las llamó alguien después que él, y ovillos neurofibrilares. los ovillos neurofibrilares adentro de las neuronas y las placas seniles, que después se supo que eran de amiloide, afuera de las células, son extracelulares. Si uno mira el cerebro de una persona que falleció con Alzheimer severo, se ve una gran atrofia comparado con un cerebro normal. Ustedes ven que las circunvoluciones están más chicas y los espacios entre estos espacios están muy aumentados de líquido cefalorraquídeo. Y si uno mira el cerebro, en este caso tau, se ve que el comienzo de la enfermedad es en la zona interna de los lóbulos temporales, que se llama temporal medial, y de ahí se va desparramando hacia anterior y posterior, y finalmente, mirado por la cara interna, compromete casi todo el encéfalo, cuando la enfermedad está avanzada. Si uno mira la resonancia de cerebro de un paciente, esto es un corte transversal o coronal se llama, y estos son los ventrículos laterales, unas como lagunas adentro llenas de líquido cefalorraquídeo, el cerebro flota en este líquido. Se ve que estos de acá son los hipocampos, este es el paciente cuando tenía 73 años, y se puede ver que estos ventrículos apenas se ve una línea sobre el hipocampo, y a medida que el paciente progresa en la enfermedad, este espacio se va agrandando a costa de una atrofia del hipocampo, que acá se ve que están bastante atróficos. En general no se usa la atrofia de los hipocampos como acá, como un signo, porque en este caso es bien evidente, pero si uno tiene un paciente solo una vez, no puede decir con certeza que está, en el caso de la derecha sin duda, porque está muy atrófico, pero si consulta a alguien así, puede tener deterioro cognitivo y todavía no reflejarse a ojo en una resonancia del cerebro. ¿Qué es lo que pasa? Estas son las placas que yo les decía y los ovillos neurofibrilares. Si esta es una neurona, o sea, estas son neuronas, estas son las placas que son extracelulares y los ovillos neurofibrilares que se van acumulando al interior de las neuronas hasta que al final queda un fantasma de pura tau y la neurona ya desapareció, queda como un esqueleto. Y la neurona puede estar cargada de ovillos neurofibrilares y seguir funcionando. Y eso es lo que da la reserva cognitiva, hasta que finalmente ya no se la puede. Y como vamos a ver más adelante, el daño que se correlaciona mejor con el deterioro cognitivo es los depósitos de tau y, más que nada, la pérdida de sinapsis o la alteración de las sinapsis, que sería el daño principal. Y este es un gráfico que muestra la evolución esto es basado en datos, pero es teórica. Y ustedes ven que aquí están los pacientes que ya tienen una demencia. Esto es tiempo. Esta es la fase que se llama deterioro cognitivo leve, cuando el paciente tiene una etapa pre-demencia, pero que pinta para. Y esta sería la etapa en que todavía es normal. Y si ustedes ven a esa altura, cuando el paciente tiene cognición normal, ya tienen el cerebro casi el máximo de niveles o casi el plató de A beta o péptido amiloide, que es el que se acumula en las placas seniles, medido ya sea en el líquido cefalorraquídeo, que es la morada, o por PET de amiloide, que es una imagen cerebral que mide la unión de un ligando radioactivo al beta amiloide y se puede visualizar in vivo. Y después viene la acumulación de tau en el líquido cefalorraquídeo. Más adelante, la atrofia, como les mostraba, de la resonancia o de fluorodeoxiglucosa que mide el metabolismo cerebral. Y finalmente, el deterioro cognitivo. Entonces se puede, como ven, uno puede tener patología de Alzheimer en el cerebro hasta 10 o 20 años antes de que se declare clínicamente la enfermedad. Se le llama, aunque yo no estoy muy convencida que sea correcto, ya se le llama enfermedad de Alzheimer, aun cuando uno no tenga deterioro cognitivo, si tiene la patología de Alzheimer. Eso está bien, yo supongo, cuando se tenga un tratamiento uno lo va a poder tratar antes de que aparezca la clínica. Ahora vamos a la pregunta que me hacían, ¿cómo se afecta la memoria en la enfermedad de Alzheimer? Primero, algunos aspectos de la memoria en general. Es el proceso en que uno codifica, almacena y recupera información. Es una capacidad que varía bastante entre individuos. Uno lo sabe, había compañeros de curso que se aprendían fácilmente un montón de cosas de memoria y otros que nos costaba bastante más. Se relaciona la inteligencia en algún grado, pero no es un ítem específico del coeficiente intelectual. Y se afecta con la edad, aunque aparentemente lo que más se afecta es la velocidad, no tanto la memoria en sí normalmente. Mucho de lo que se sabe de la memoria actualmente proviene de los estudios de un paciente HM. Por muchos años se le llamó HM para proteger su identidad, pero ahora se sabe, su nombre se me olvidó ahora, porque ya falleció. Vivió entre 1926 y él a los 26 años o a los 23, no estoy segura, le hicieron una cirugía de la epilepsia porque tenía una epilepsia intratable que no lo dejaba ni estudiar ni trabajar, y le sacaron los polos anteriores de los lóbulos temporales, que incluyó más o menos este es el hipocampo en el cerebro, más o menos los 2/3 anteriores y los 2 polos del hipocampo. Quedó sin epilepsia, o una que otra al año, pero quedó con una amnesia anterógrada muy severa, o sea, él no podía aprender nada nuevo. Unos años, el mismo cirujano que quedó muy afectado por esto y escribió que nadie más hiciera esta cirugía para la epilepsia, él describió después el caso del paciente y una neuropsicóloga, y él era muy afable, muy alegre, muy solícito, siguió colaborando con la investigación por años, hasta que falleció. Sin embargo, si la neuropsicóloga salía a la pieza y volvía a entrar, él no se acordaba que ella había estado antes. Entonces, él quedó con esta memoria anterógrada, o sea, no puede aprender nada nuevo, y retrógrada, que abarcaba más o menos 2 años, y las cosas que le habían ocurrido en la infancia las tenía mejor grabadas que las más posteriores, o las más cercanas al momento de la cirugía. Lo interesante que describieron con él, acá ustedes ven cómo le quedaron sin hipocampos en el corte coronal de su cerebro. Que él tenía la memoria de trabajo conservada. Y ahora vamos a ver qué es lo que es la memoria de trabajo, pero es, por ejemplo, si uno le dicen un número de teléfono y va a la otra pieza a discarlo. Eso que vendría a ser como la memoria RAM del computador, lo que uno le queda en la cabeza para usarlo inmediatamente. La memoria procedural, que es otro tipo de memoria, es la memoria que uno no hace pensando, sino que la hace intuitivamente o porque ya la aprendió, también la tenía conservada. Y eso se demostró en que le enseñaron a dibujar mirando un espejo, o sea, mirando solo el espejo, dibujar la estrella por dentro de la marca, que es algo motor bastante difícil, y que él no se dio cuenta, pero porque no se acordaba que lo había hecho antes, cada día que lo hacía y la repetía, fue aprendiendo a hacerla mejor. Y ahí dijo, oh, esto ha sido más fácil de lo que yo esperaba, porque al final lo hacía bastante bien, pero él no se acordaba que lo había hecho. Entonces, en base a todo lo que se aprendió de él, se describe o se empezó a clasificar la memoria en declarativa o explícita y no declarativa o implícita. Esta es toda la memoria que uno aprende sin pensar, sin, por ejemplo, los hábitos, el priming, si a uno le dan una serie de palabras y después le dicen que escoja cuáles oyó antes, uno tiene más frecuencia de decir las que escuchó hace poco. Condicionamiento clásico, el de Pavlov, etc. En cambio, la declarativa es la de los eventos o de los hechos que ocurrieron poco antes o bastante antes. Esa se llama explícita. Entonces, como les decía, hay una etapa de codificación o registro, otra etapa de almacenamiento y otra etapa de evocación o recolección. Y ahora se sabe que son funciones diferentes y que ocurren en distintos lugares, por eso se pueden separar sus alteraciones. Y otro tipo de clasificación de la memoria la llaman inmediata o la memoria de trabajo. Acá hay un poco de confusión porque la gente la llama de corto, algunos investigadores la llaman de corto plazo como equivalente a la inmediata, lo cual parece no ser lo correcto porque la inmediata o la memoria de trabajo, que es esta tipo RAM, la de guardar un número de teléfono, etcétera, no sería del lóbulo temporal, sino más bien de las funciones del lóbulo frontal, de la función dorsolateral del lóbulo frontal, que es más una función ejecutiva. La memoria reciente, que sería la de corto plazo, qué es lo que se hizo el día anterior, qué almorzó, etc. Y la memoria de largo plazo, que se llama a las cosas que uno, o la memoria semántica, son las cosas que uno aprendió de siempre, por ejemplo, cuántas patas tienen los mamíferos, de qué color es una manzana, o hechos biográficos, cosas que a uno le pasaron en el tiempo anterior. Y esta es como se clasifica más recientemente, que le llaman memoria episódica, como recordar una lista corta de cosas, que se desayunó, etc. La memoria semántica, que era esta que les decía, como el primer presidente, el color de una manzana. La memoria procedural, que es como esta implícita, como andar en bicicleta, aprender una secuencia de número de teléfono sin saberse los números, sino saber solo la secuencia motora. Y la memoria de trabajo, que es esta que es parte de la función ejecutiva. ¿Y cómo se localiza en el cerebro las distintas memorias? La semántica se localiza en el lóbulo temporal lateral e inferior. La procedural tiene que ver con el cerebelo, con los ganglios basales y el área motora suplementaria. Y la working memory, o la memoria de trabajo, que es de los lóbulos frontales y que tiene que ver más con la función ejecutiva, no tanto con la función de la memoria. Y la memoria episódica, que es la que se afecta en el Alzheimer y la que se estudia ahora para tratar de entender lo que pasa, es la memoria, se localiza principalmente en el hipocampo, pero no solo en el hipocampo, sino en la zona perihipocampal, que incluye la corteza entorrinal y el subiculum, el fornix, los cuerpos mamilares, el núcleo talámico anterior, el núcleo y la corteza cingulada y el lóbulo frontal. Y esto conforma el circuito de Papez. Todas esas zonas intervienen en la memoria episódica. Entonces, para tener una buena memoria de lo que tomó de desayuno, de lo que se hizo hace un rato, requiere una indemnidad del hipocampo y de las regiones vecinas. Sigue la ley de Ribot, se dice, que uno lo ve mucho en los pacientes, los eventos que ocurrieron junto antes, o sea, hace poco, son los que no se recuerdan. Los más antiguos están mejor recordados. Entonces es muy frecuente que los familiares digan, no, si mi mamá tiene muy buena memoria, se acuerda de cuando era chica, de cuando fue al colegio, pero no se acuerda nada de lo que recién hizo. Incapacidad de aprender nueva información, que es la amnesia anterógrada, y la incapacidad de evocar, la información aprendida recientemente, que es retrógrada de corto plazo. En cambio, la remota generalmente está indemne. Y los lóbulos frontales, como les mostraba en el gráfico, también son necesarios para la memoria episódica. Entonces, sería análogo al junior. El lóbulo frontal haría la pega de agarrar la información y archivarla en el lugar correcto. En cambio, la memoria, el lóbulo temporal medial se encarga de guardar, es el cajón donde está la memoria, que después de un tiempo se va a zonas de corteza, que ahí es donde está la memoria de largo plazo, la memoria ya más semántica, que como que la información más antigua se guarda allá en otros lugares que no es la zona perihipocampal. Entonces, si falla el lóbulo frontal, frontal puede ser difícil, pero no imposible acceder a la información. Si al paciente uno le dice, a ver, era un color, ¿cuál color era? Y el paciente se acuerda, porque se grabó. En cambio, el paciente que tiene Alzheimer, uno le dice, ¿cuál color? Que era un color, y dice, ¿un color? Y empieza a mirar al familiar para que le ayude, porque no tiene idea de que yo le dije antes que le iba a preguntar por un color. Entonces la información en el lóbulo, cuando falla el lóbulo frontal, que se puede ver en la depresión o se puede ver en las demencias vasculares, o cuando uno está inatento, se queda mal archivada, entonces en el momento de acceder a ella no se logra. En cambio, cuando es falla de grabación, no se recupera ni con claves. Si el lóbulo temporal está dañado, la información no se grabó, no se puede recuperar. Pero sí la antigua, de meses o años, porque se ha guardado en zonas del cerebro que están indemnes y que no tienen el circuito de papel dañado. Entonces, como se ve, con el paciente deprimido o con Alzheimer, los dos pueden tener falla de memoria, pero con el depresivo falla el junior, entonces con las claves lo puedo acceder, con el Alzheimer ni con las claves se puede acceder. ¿Y qué sucede entonces en el Alzheimer? Ahora, como les dije antes, existen ligandos para A-beta y para tau, que son radiactivos, y entonces con un PET scan se puede visualizar el depósito de amiloide in vivo, que antes solo se podía hacer con una biopsia cerebral, que obviamente no se hacía, o post-mortem en el estudio anatomopatológico. Y aquí se ve entonces que para amiloide el depósito es frontal y temporoparietal en la cara interna, pero no específico en los lóbulos temporales. En cambio, para tau, si ustedes ven, en el lóbulo temporal medial, en los temporales laterales y frontal. Entonces, abajo es cognitivamente normal. Este es un paciente es una forma de Alzheimer que se llama atrofia cortical posterior, y este es con el paciente que tiene una forma, el Alzheimer típico, que es el amnésico. Y ustedes ven cómo empieza acá y el lóbulo temporal. En cambio, en el amiloide es más difuso el depósito. Y el tau se sabe desde el tiempo de la anatomía patológica que la correlación entre alteración cognitiva y depósito es mejor con tau que con amiloide. Y con eso, como les decía, como ya se sabía de estudios anatomopatológicos, se correlaciona mejor tau, y estas alteraciones, como se veía en el otro gráfico, ocurren 10 a 20 años antes de que empiece la clínica. Para el A-beta, que tiene menor correlación con la clínica, se sabe que se deposita primero en zonas del precuneo. A ver si lo muestro. El precuneo es más o menos esta zona. Aquí. Esta es amiloide, que es una zona que tiene que ver con como una zona de interconexión de muchas áreas. Y eso ocurre precozmente, es como de los primeros lugares donde se deposita. Entonces, ¿cuál es el daño en Alzheimer? Que se sabe. Se creía antes que lo tóxico del beta-amiloide eran las placas de amiloide, los patacones que se veían extracelulares. Después se pensó que eran las fibras lo que era tóxico. Y finalmente, lo que se acepta ahora es que son oligómeros, o sea, entre 2 y hasta 12 monómeros de A-beta juntos, que serían los tóxicos, y que la toxicidad sería a nivel sináptico. Tau fosforilado, porque lo que sucede con tau es una hiperfosforilación en varios sitios, y cuando tau, la función normal de tau es mantener estables los microtúbulos que permiten el transporte de material desde el soma de la neurona hasta las terminales sinápticas, que a veces están metros de distancia. En el cerebro no, pero en el resto del organismo pueden ser bien largas. Y si tau está fosforilado, se desune de los microtúbulos y estos se disgregan y se pierde la capacidad de transporte axonal. También se sabe que hay disfunción sináptica, que lo vamos a ver en un minuto, disminución del número de sinapsis y muerte neuronal finalmente. Y también se ha descrito daño por inflamación, daño vascular, daño por, daño por radicales libres o oxidación y disfunción mitocondrial. Se cree que muchos de estos daños son precoces, antes, en etapas más iniciales de la enfermedad, pero todavía no está claro. Si esta es una representación de la potenciación post-tetánica, o long-term potentiation en inglés, se sabe que los oligómeros, se sabe ya desde el año 98, que in vitro si uno en cortes de hipocampo administra oligómeros de A-beta, se interfiere con la potenciación post-tetánica, que es, o sea, con la long-term potentiation, que es una facilitación sináptica que ocurre después de una estimulación tetánica, y que se supone que se homologa a la memoria, a cómo serían las bases moleculares de la memoria. Se sabe también que los oligómeros se unen a receptores de NMDA, que están muy implicados en la potenciación, en la long-term potentiation, y se une a proteínas de maduración dendrítica, de diferenciación y de creación de espinas, lo que habla de que por qué interfiere en la sinapsis. Acá quiero hacer un honor al doctor Luco, que fue mi maestro, y sus experimentos. Estos los publicó en Nature en el año 64-66. Yo todavía no llegaba a su laboratorio, no creen que soy tan vieja. Y él, mucho antes que Candel, hizo estos experimentos en cucaracha, en que les cortaba las patas delanteras a la cucaracha, que normalmente la cucaracha usa sus patas traseras para, con la pata anterior, limpiar las antenas. Entonces él le cortaba las patas delanteras y la cucaracha al principio estaba bien torpe, pero después de un tiempo lograba pararse en 3 patas y con la pata del medio limpiar la antena. Y lo que Luco demostró era que, voy a correr esto porque me interfiere, ahí él demostró, poniendo un estímulo acá y el registro en el ganglio de la pata más trasera, que las sinapsis que normalmente estaban cerradas cuando la cucaracha, el paciente, cuando la cucaracha tenía todas sus patas, al cortarle las patas traseras y tener que aprender a usar las del medio para limpiarse, se facilita esta sinapsis que normalmente estaba cerrada, o sea, se abre una sinapsis. Y eso se lo publicaron en Nature en 2 artículos, súper interesante como modelo de cómo la sinapsis y la función sináptica está involucrada en la memoria. Ahora unas pocas palabras de la neuroinflamación en Alzheimer. Se sabe que hay inflamación y proliferación de microglía y de astrocitos alrededor de las placas y difuso en el cerebro. También se sabe hace menos tiempo que una mutación en una proteína que se llama TREM2, que es producida por la glía, es un factor de riesgo de Alzheimer. Así que ha cobrado interés últimamente todas las alteraciones que ocurren en el sistema de inflamación e inmunidad durante el Alzheimer. Se sabe que tanto tau como A-beta activan la neuroinflamación. Después se sabe que la microglía y los astrocitos tendrían, no está todo muy claro, pero tendrían un valor, un efecto beneficioso en etapas iniciales, porque ayudan a fagocitar a beta y tau, también liberan neurotrofinas para favorecer el crecimiento sináptico, etcétera, y eventualmente de neuronas, pero más tardíamente, cuando la enfermedad avanza, se harían deleterios liberando sustancias inflamatorias, alterando la función sináptica, interfiriendo con la eliminación de beta y de tau, etc. Perdón. Y quería contarles solo brevemente en qué estamos trabajando nosotros. Hemos descrito hace ya unos años de que hay una asociación inversa entre cáncer y Alzheimer, en que los pacientes que han tenido un cáncer tienen menos riesgo de desarrollar Alzheimer y los pacientes que tienen Alzheimer tienen menos riesgo de desarrollar cáncer en el futuro. Y planteamos que puede haber un mecanismo biológico común que esté desregulado en sentidos opuestos. Entonces, nuestra investigación actual es, queremos probar si estimulando la inmunidad, que sería beneficiosa, podríamos proteger del desarrollo de Alzheimer. Entonces, lo que vamos a hacer en un proyecto FONDEF es inyectar una vacuna de lisado de células de melanoma que se llama Trimelvax, que fue desarrollada por Mercedes López y Flavio Salazar en la Universidad de Chile, y que ellos han demostrado que estimula la inmunidad para defenderse de un tumor en pacientes y en animales. Nosotros queremos probar si esta misma vacuna que estimula esta inmunidad podría ser útil para prevenir el desarrollo de la patología de Alzheimer en un ratón transgénico. Eso no les puedo contar los resultados porque todavía no los tenemos. Entonces, en resumen, no está claro cuál es el mecanismo íntimo de la falla de memoria en el Alzheimer, pero el hecho de que tau se acumula en la zona temporal medial, que es similar a lo que ocurre en el paciente HM, y que se afecta la función sináptica del hipocampo, todo hace pensar que en la zona temporal medial está involucrada. Pero también es posible pensar que la alteración en un sitio que está interconectado por axones o dendritas con otras zonas podría alterar la sincronización de redes y de esta forma interferir con el proceso normal de la memoria. Por ejemplo, en el cingulado posterior y el precunio, como yo les decía, que es una región donde se acumula precozmente amiloide, es un hub de interconexión y podría explicar que inicialmente esté alterado. Y finalmente que tau y A-beta están involucrados sinergísticamente en producir daño, junto con neuroinflamación, oxidación y etc. Y quería agradecerles a los chicos que trabajan conmigo, a la colaboración con Mercedes, Flavio Salazar, Alejandra Gleisner, Fabián Tempio, y al financiamiento que tenemos de FONDES y de FONDEF IDEA para desarrollar esta investigación. Muchas gracias.
Thank you very much, Professor Berens, for an excellent talk. I think it's a good way to finish, because we now understand how all the things we've seen in the previous day translate into memory deficits in real human persons. I will switch to Spanish just to honor the presentation. Muchas gracias a la doctora María Isabel Berens por su excelente charla. Vuelvo a repetir, es un honor tenerla con nosotros. Yo la conozco en persona, conozco ese fondo de pantalla con los ladrillos fiscales, y estoy muy agradecido y muy feliz de que hayas aceptado nuestra invitación, y tu charla fue muy, como dije en inglés, una excelente manera de redondear todo lo que vimos desde los aspectos más neurobiológicos a cómo esto se traduce en personas, en humanos reales finalmente, y en patología médica. Yo tengo una pregunta que quizás le nace a muchos. Señalaste al inicio que el Alzheimer es una enfermedad muy importante, que ha ido aumentando su prevalencia, sobre todo a medida que la expectativa de vida de los seres humanos ha ido aumentando. Y me nacen dos preguntas al respecto. Uno, ¿por qué no se considera una parte normal del envejecimiento? Porque si uno mira las cifras sobre 80 años, las personas que tienen algún grado de Alzheimer, no es poco. Entonces uno, ¿en qué momento dice, bueno, pero esto no es? la mayoría de, esto no es las consecuencias de envejecer. Y dos, también bastante obvia, ¿hay alguna manera o alguna terapia, ya sea desde la cosa más farmacológica o médica hasta entrenamiento, hacer sudoku, que se ha visto que ayude a las personas a, si no mejorarse, a por lo menos enlentecer el desarrollo de la enfermedad?
A ver, respecto de la primera pregunta, se planteó durante mucho tiempo que era parte del envejecimiento normal, pero hay personas que fallecen sin tener depósitos de amiloide ni tau en el cerebro y con la cognición normal. Entonces, y no son, o sea, son raros, pero no son uno que otro, son una buena cantidad, no sé los números en este minuto. Entonces, se piensa que es más bien una patología adquirida asociada a la edad y no un equivalente de enfermedad. Pero eso no te puede decir con certeza. Si uno mira por cohortes, estudiando qué cosas cambian con la edad, es difícil decir porque son poblacionales, entonces uno no sabe exactamente si esos pacientes que se están siguiendo año a año, en qué minuto empezaron a desarrollar la enfermedad y entonces empiezan a ensuciar la muestra. Entonces, no está claro qué es lo que sí se altera por la edad y qué es lo que no tiene que ver con edad y es parte de una enfermedad. Pero lo que se acepta ahora es que son patologías, no es normal tener Alzheimer. Respecto de qué se puede hacer para prevenir y de tratamiento, yo no lo toqué acá, pero es válido. Existen dos aprobados por la FDA, tratamientos que modulan neurotransmisores. Lo primero que apareció fueron los anticolinesterásicos, y eso se basó en que el núcleo de Meynert era uno de los primeros que se degeneraba, entonces se planteó que podía y resultó, pero no es un efecto espectacular, dura unos 3-4 años en que la curva se enlentece, el paciente con anticolinesterásicos progresa más lento que el sin, pero después se hacen paralelas. Y se sigue, o sea, no es curativo ni es neuroprotector, tiene un efecto como de estimular la alerta, porque es anticolinesterasa, favorece la transmisión colinérgica, el paciente está más despierto, más participativo, pero no, en las pruebas de memoria específicamente no hay mejoría. Y el otro tratamiento es un modulador del receptor de NMDA, de receptores de glutamato, la memantina, que en teoría sería evitaría la excitotoxicidad, modularía la entrada para evitar el exceso de calcio intracelular y desencadenar la excitotoxicidad. Por temas de mercado, la memantina se probó y se aprobó para Alzheimer moderado a severo porque ya existían los anticolinesterásicos para el leve a moderado. Pero si uno piensa que es un neuroprotector su mecanismo de acción, es absurdo esperar a que la enfermedad esté avanzada para iniciarlo. Así que en general se usan precozmente los dos. También hay otro fármaco aprobado, pero no por la FDA, que es un lisado de cerebro de porcino que se postula que actuaría como nutrientes para la reparación del tejido cerebral. Se inyecta endovenoso, cruza la barrera hematoencefálica, porque son fragmentos pequeños. Algunos pacientes, más bien los familiares, dicen que sí les hace efecto, así que a veces yo lo uso, pero en otros no, si no sirve, se suspende. Eso sería en cuanto a tratamiento. Ahora, en cuanto a prevención, tampoco hay fármacos, pero sí hay evidencia de factores que intervienen. La educación es un factor importante, influye o sea, se calcula, si uno corrigiera la educación, disminuiría en 8% el riesgo de tener Alzheimer de la población. Parece que es la educación a edades bien tempranas más que la otra, pero se recomienda mantener, adquirir conocimientos nuevos a lo largo de la vida. Después están los factores genéticos, que eso no se pueden modificar, como tener APOE4, tener mutaciones, etc. Y hasta un 45% podría ser modificable con la educación, como decía, con la audición. Si hay problemas de audición, interfieren un 9%, que es bastante, y después están todos los factores de riesgo cardiovascular, como hipertensión, obesidad, diabetes, sedentarismo, tabaco, alcohol, TEC, etcétera. Varias cosas que si uno las evita durante la vida, previene la demencia en general, no solo de Alzheimer, la demencia en general.
Muchas gracias. No sé si son buenas noticias después de la pandemia, en que hemos visto que los consumos de sustancias han aumentado bastante. Yo creo que además el encierro no favorece el aprendizaje. Bueno.
Ni el ejercicio, que también no lo mencioné, pero el ejercicio es uno de los factores importantes para prevenir demencia.
Ya, tengo algunas preguntas del público, doctora Berens, que me parecen. Una es, alguien pregunta si el rol de la vitamina B12 tendría un rol en la aparición de enfermedad de Alzheimer, y qué rol cumple dicha vitamina en los procesos sinápticos, si se sabe algo de que sea necesaria para, no sé, aumentar dendrina.
Interesante pregunta. Si tiene efectos sobre la sinapsis, no lo sé. Interesante investigarlo. En la literatura sí está. Y respecto de interfiere, pero no estoy segura que sea por el Alzheimer. Yo creo que es por la memoria en general, o demencia en general, interviene. Digo porque cuando uno ve pacientes con déficit de B12, tienen fallas de memoria, pero probablemente son secundarias a fallas de la función ejecutiva. El paciente está más inatento, o está más indiferente, más apático. Entonces, pero habría que explorarlo mejor para estudiar en qué específicamente, si es en la memoria episódica o no, el efecto, y si es a nivel sináptico o no. No tengo la respuesta.
Buena respuesta. Hay una pregunta respecto a cómo aumentar la inmunidad, y yo aprovecho de colgarme esa. No solo aumentar la inmunidad, veo que ustedes están intentando hacerlo con un mecanismo de la vacuna, pero sí, además de cáncer, otros trastornos del sistema inmune, como la enfermedad autoinmune, se han visto o causales o protectores de demencia, y en particular de Alzheimer.
Hay unos datos antiguos de la literatura que retrospectivos, que los pacientes con artritis reumatoidea desarrollan menos Alzheimer. Y se pensó que era por el tratamiento que recibían durante muchos años de antiinflamatorios. Y se hizo un ensayo clínico en pacientes administrando antiinflamatorios ya con la enfermedad empezada, digamos, en distintas etapas, y no hubo beneficio. Entonces quedó la duda si era porque se hubieran administrado anterior al desarrollo de la enfermedad, hubieran sido positivos, pero tampoco eso está claro. Y lo que yo he leído últimamente es que a lo mejor la misma inflamación que produce la artritis reumatoidea podría ser, en algunos papers lo leí hace poco, que también se plantea que podría ser lo que está defendiendo del Alzheimer, y no el tratamiento antiinflamatorio.
Gracias, Lisi. Otra pregunta, un poco clarificadora, porque me preguntan sobre si la demencia senil está relacionada con el Alzheimer o no, y qué diferencias hay, o solo hacer una clasificación de nuevo para que la gente quede claro.
A ver, demencia en general, dije yo, es la pérdida de las capacidades cognitivas que afectan las actividades de la vida diaria. Es un término muy amplio. O sea, una persona con una enfermedad de Down, con un retardo del nacimiento, puede desarrollar un Alzheimer. De hecho, neuropatológicamente la desarrollan todos después de los 40 años, y puede clínicamente empeorar y se llama demencia igual, porque de su nivel bajó a otro. Las causas de la demencia son muchas y el Alzheimer es la más frecuente, pero no es la única. Está la demencia con cuerpos de Lewy, que es una demencia que se asocia a parkinsonismo, a alucinaciones visuales y a fluctuaciones en la cognición. Y se puede parecer al Alzheimer, pero no es clínicamente igual, y patológicamente es diferente porque se acumulan no amiloide y tau, no beta amiloide y tau, sino alfa-sinucleína y unos cuerpos de Lewy se llaman. Después está la demencia frontotemporal, en que se afectan en el Alzheimer se afecta el lóbulo temporal medial y las regiones parietales y el precuño, como yo les decía, que es esta parte de acá. En la demencia frontotemporal se afectan los lóbulos frontales y los temporales, a más anteriores, y dan trastornos de conducta por el lóbulo frontal o alteraciones del lenguaje por las fallas de los lóbulos, especialmente si es izquierda. En el Lewy se afectan las áreas más occipitales o difuso. Entonces, depende de la zona del cerebro que se afecte, la clínica que va a dar, y depende de qué proteínas se asocian a qué patología pertenece. Entonces, hay un Alzheimer que se llama cortical posterior, en donde se deposita en la zona occipital mucho amiloide y beta, y es una de las formas atípicas de Alzheimer porque parte en una zona que no es la habitual, que es la de la memoria, y hay otro Alzheimer que se llama así porque si uno mira el cerebro, tienen amiloide y tau, y ese se deposita en la zona frontal y se confunde con la demencia frontotemporal, que es la otra variante. O sea, no sé si respondo a la pregunta, pero depende de la zona del cerebro donde parta la enfermedad, y depende de qué proteínas se acumulan, la que uno lo defina como una u otra.
Muchas gracias. Nos estamos llenando de preguntas, no sé si vamos a alcanzar a responderlas todas. Tengo aquí una de la doctora María Cecilia Hidalgo, me imagino. Supongo que será ella, para los que no la conocen, es Premio Nacional de Ciencias y profesora de la Facultad de Medicina de la Universidad de Chile. Me está preguntando si el consumo de antioxidantes naturales puede contribuir a prevenir la enfermedad de Alzheimer.
Hay muchos estudios en animales transgénicos y estudios a nivel celular, etcétera, que sugieren que los antioxidantes son útiles para prevenir. Pero desgraciadamente los ensayos clínicos en pacientes no han podido demostrar diferencias significativas entre placebo y tratamiento. Pero eso puede ser por lo que siempre pasa con los ensayos clínicos, el efecto placebo, donde el paciente solo se mejora porque cree que está tomando algo que le hace bien. Entonces mejora también el placebo y mejora pero por errores y por las diferencias y las variabilidades entre pacientes, no se logra demostrar el efecto de los antioxidantes. Entonces, esa respuesta clínicamente todavía está pendiente. Se usan, pero como off the label, sin evidencia dura todavía, pero como pueden no, o sea, la mayoría no son dañinos, se usan, pero no como una de las indicaciones formales de tratamiento.
Existen algunas preguntas respecto al tratamiento, por ejemplo, algunas de oligómeros, pero prefiero decir que, por lo que tú señalaste, los únicos tratamientos autorizados son los que ya señalaste, que tendrían como target el sistema colinérgico y los moduladores de receptores NMDA, si no me equivoco. Una que yo creo que puede englobar varias son, ¿existe alguna idea de los mecanismos que el ejercicio echa a andar que de alguna manera protejan contra el desarrollo de la enfermedad de Alzheimer?
Sí, se plantea que puede ser uno que mejore lo cardiovascular, mejore la irrigación cerebral, etc. pero la otra es que se basa más en estudios en animales de ratoncitos que hicieron ejercicio con esta ruedita y los que no. Tienen más neurogénesis los ratones que hicieron ejercicio comparados con los que no hacen ejercicio, entonces se plantea que sí, que interfiere en el crecimiento de células espinas dendríticas, mayores números de sinapsis y neurogénesis, mayor número de neuronas. Pero eso in vivo no se ha demostrado en pacientes que sirva efectivamente por el ejercicio. Pero sí hay datos de que el ejercicio es útil en la prevención, como les decía antes. Y ahí sirve el ejercicio tanto aeróbico como el ejercicio de caminar o el ejercicio de hacer el aseo en la casa. Hay datos de que o sea, no estar sentado sería lo que ayuda, o no estar acostado todo el día.
Muchas gracias. Voy a dejar la última pregunta, Lizzy, porque en verdad, si no vamos a terminar, solo Dios sabe cuándo, y esto hay que finalizarlo. Muchas gracias a la gente que está preguntando y a ti, porque eso significa que la gente se entusiasmó con la presentación. Otro tema de moda es la microbiota intestinal, que hoy día se sabe que juega un rol en el sistema inmune y están saliendo muchos papers y trabajos que también hay una interacción entre intestino y cerebro. ¿Qué se sabe de microbiota intestinal y demencia en general?
Sí, ha estado como muy novedoso últimamente, y yo no sé exactamente cómo podría ser el mecanismo pero se plantea que existen neurotróficos que secretan las bacterias que atraviesan la pared intestinal y llegan vía sanguínea al cerebro y que estimularían, pero yo no sé los detalles como para contestarlo también como se merece la pregunta, pero es un área muy interesante de investigación y que todavía no tenemos la última palabra, pero para una enfermedad que tiene hasta ahora tan poco tratamiento, el último, el de la memantina, el que actúa sobre los receptores de NMDA, se aprobó en el 2003. Y por la FDA no hay ningún nuevo tratamiento para la enfermedad de Alzheimer hasta ahora. Existe una vacuna que se llama aducanumab, que es un monoclonal antiamiloide, que se supone que la FDA va a resolver ahora en mayo, lo tenía que ser en marzo, se postergó para mayo, y hay toda una discusión porque en los ensayos clínicos tenían una parte, los resultados no mostraban diferencias, pero en un subgrupo de pacientes sí mostraron diferencias, así que ojalá, uno quiere que salga algo para poder ayudar a los pacientes, pero todavía eso no existe y esperamos que sí salga pronto. Y por eso conocer más de todos los mecanismos donde fuera posible desarrollar una nueva terapia, así como la microbiota, es muy importante.
Muchas gracias, profesora Berens. En verdad una charla bastante interesante, muy entusiasmante. O sea, el público sigue haciendo preguntas. Lamentablemente esto no puede durar eternamente. Dejo invitados a aquellos que tienen preguntas a hacérselos directamente a la doctora Berens o a nosotros, y nosotros se las haremos llegar. Ahora nos corresponde hacer una sesión de cierre del simposio. Le voy a pedir a la doctora Berens que por favor nos deje de transmitir su pantalla para poder pasar a la siguiente pantalla. Voy a hacer unas palabras cortas. Yo creo que ha sido muy interesante esto. Nosotros estamos muy contentos, después yo haré algunas palabritas, pero antes quiero dejar a Nuik van der Ver, que es...
¿Cómo desconecto? No me resulta.
¿Cómo desconecto? ¿Te podemos desconectar, yo creo?
Ya.
Ahí te desconectaste, puedes apagar tu micrófono. Estamos... como iba diciendo, ahora quisiera dejar con ustedes a la profesora Nuik van der Ver de la Universidad de Santiago de Chile, que quisiera dar unas palabras al cierre de este evento, ¿no? Es todo tuyo.
Muchas gracias, José. Sí, en verdad, me alegro mucho ver la gran conectividad y la actividad de las preguntas y respuestas también hasta esta última sesión, que sabemos que han sido 4 días bien largos de interacción, de compartir de muchas ideas, muchas novedades también, y ese espacio es justamente lo que nosotros, al menos de parte de la Universidad de Santiago, quisiéramos generar. Entonces esperamos que haya sido un espacio importante en ese sentido para el intercambio de ideas, de compartición de investigación y de áreas específicas de cada uno. Yo quisiera partir este cierre con algunos datos interesantes tal vez para el público, y también como en agradecimiento a todas las personas que participaron en este simposio de Neurosur 2021, donde, bueno, en primer lugar, en verdad agradecer mucho a los 4 moderadores que tuvimos, que un trabajo arduo y bien importante para hacer que todo fluya y que tengamos, digamos, un hilo conductor entre una presentación y otro, más allá de la logística, en la comprensión de las temáticas y la síntesis de las ideas. Y por lo tanto, un agradecimiento especial para Francisco Flores, Elías Leiva, José Egaña y Antonella Pena también, que ha sido clave, digamos, en el éxito del simposio. Aparte, contamos con un total de 16 panelistas que expusieron, de instituciones participantes de diversos países, de Estados Unidos, Canadá, Chile principalmente también, de parte de 3 universidades nacionales también, y por ende agradecer principalmente al Instituto Tecnológico de Massachusetts, Massachusetts General Hospital, Harvard Medical School también, la Universidad de Harvard en general, la Universidad de Toronto, que también estuvo presente, y particularmente las 3 universidades chilenas, la Universidad de Chile, que tuvimos alegrado de la última presentación justamente, Pontificia Universidad Católica de Chile, y también la Universidad de Valparaíso, que estuvieron las 3 presentes en este simposio importante. Y claramente también agradecer todo el apoyo de parte del Instituto de Neurociencia Biomédica, que ha sido clave para el éxito del evento. También en cuanto a números, hemos sido impactados por la cantidad de interés, por lo cual pudimos contar en el día 1 con un total de 390 espectadores, en el día 2 más de 300, y en el día 3 con 242 participantes, lo cual nos hace un total de inscritos por el evento en generalidad de más de 500. Por ende nos alegra mucho, digamos, encontrarnos con un público que hace eco de las temáticas que se hablaron, de lo importante, lo relevante que son para el avance de la ciencia en los ámbitos que se han ido conversando, y también de la cantidad de países. Tuvimos una totalidad de más de 10 países conectados a la totalidad de las exposiciones, de Argentina, Chile obviamente, Brasil, Canadá, Estados Unidos, Honduras, México, Nicaragua, Portugal, Suecia y Uruguay. Así que eso también nos demuestra, digamos, la conectividad, y sobre todo en un contexto de pandemia, este tipo de actividad nos demuestra la importancia de poder seguir avanzando y colaborando a nivel internacional, ya que la fortaleza de la cooperación internacional está justamente en buscar nuestra complementariedad, el apoyo, digamos, mutuo entre investigadores y académicos, médicos también en estas áreas, para aprovechar, digamos, de seguir avanzando, estrechando lazos y fortaleciendo las redes de colaboración en las diferentes áreas que existen en esta temática. Así que sin nada más que agregar, si es que me faltó alguien en mis agradecimientos, la verdad, siéntanse por favor nombrados también, y espero que más que nada haya sido muy provechoso para todos los participantes, y que podamos, bueno, espero que se hayan hecho también nuevos nexos y que pueden seguir en contacto para seguir colaborando de aquí al futuro. Así que de parte de la Universidad de Santiago, al menos, un agradecimiento tremendo a tanto participantes como expositores, y espero que sigamos en contacto por todos los vías que tenemos disponibles en estos tiempos virtuales. Muchas gracias, José.
Gracias a ti, Anu. Voy a compartir pantalla, ¿o no? Espérense. ¿Algo malo hice? ¿Me ven? Yo no me veo.
Sí lo
Perdón.
vemos.
Ya, después de ese error, yo quiero ser breve, solo decir que para todos los que están pensando preguntándose cómo nace este tipo de iniciativa, lo primero que les decirles, y lo más importante, es que esto es para nada el esfuerzo de uno o unas pocas personas. Este es un esfuerzo colaborativo entre varias personas y varias instituciones. Y dentro de las instituciones que yo quisiera agradecer, y a nombre del comité organizador, está la Universidad de Santiago de Chile, la Universidad de Chile, la Universidad de Harvard y el MIT, que han sido, diría, los 4 pilares en términos de organizaciones o en términos universitarios que nos han ayudado. Un especial reconocimiento al Instituto de Neurociencias Biomédicas, el BNI, a la Caro Cubillos, a Bernardita, a Topia, un poco por ellos, que han sido fundamentales para esto. También agradecer en lo personal a la ANID. Parte de los fondos que cubren estas actividades fueron proporcionados por la ANID en un fondo Redes 2018. Y en un tema más personal, yo quisiera partir por agradecer a la audiencia. Todo estos esfuerzos y ganas se ven recompensados cuando la gente demuestra interés, y el objetivo de acercar la neurociencia de primer nivel no solo a los chilenos, sino que en particular a un público latinoamericano. Luego de escuchar los números de ANUC, creemos que se ha cumplido, y entonces vaya para todos los que nos escucharon y escucharon a los panelistas, mis más sinceras gratitudes. No puedo dejar de agradecer a nuestros speakers. Tuvimos speakers de primer nivel todos los días, y eso se agradece. No siempre se puede contar con gente de primerísimo nivel hablando de los temas que lo apasionan, y en esta oportunidad tuvimos no uno ni dos, sino que tuvimos muchos speakers de primerísimo nivel. Y finalmente, aún más personal, quisiera agradecer a mis amigos, que son el doctor Flores, el doctor Leiva y el doctor Pena, con los 4 que hemos empujado para que esto salga adelante. Muchas gracias, el esfuerzo creo que ha valido la pena. Y nada, pues los dejo, con eso termino, y los dejo invitados a el taller, a aquellos que vayan a ser seleccionados la próxima semana. Muchas gracias. Voy a despedirme entonces, se me olvidó, mal educado. Muchas gracias nuevamente, aquellos que están todavía en línea, muchas gracias por haber venido.